disease, infection, then problem associated with food spoilage, beneficial aspect, right? All kinds of stuff we learned. And now let's say at least control and prevention. When you talk about control and prevention, that means we are going to talk about mostly where we have undesirable property, where biofilm can cause problem, whether it's a product development, product safety, or shelf life, and also from the disease standpoint, if these organisms or biofilm can be eradicated or eliminated, and from the standpoint of therapeutic or clinical application, we have to know how to prevent or control, right? So I'll give you some bigger, again, like big pictures, some detailed information, but then you can read up a little bit more how specifically some of those things are done. Okay, again, same picture you've seen again, just to remind you what is, how they form, and what are the final steps, and of course, what are the different factors. Again, when you try to control biofilm formation or also prevent biofilm formation, you have to take into account how do they are formed, right? Why they attach, how they attach, right? All of those factors, and what do they do during biofilm formation, right? Again, all of those things we talked. Now you need to apply it, and that knowledge is going to be extremely important for our strategy. Exopolysaccharides, right? Polysaccharide, exopolymer, polysaccharides, proteins, eDNA, lipids, they are the major matrix molecule that hold these molecules together. Then, of course, the quorum sensing. So, if you know what makes these biofilms work, then these are the certain approaches you can undertake to help prevent or control biofilm formation. Okay. Throughout this lecture today, we're going to talk about individual steps, right? So if you just broadly take a look at it, say, can we use antibiotics or adjuvant agents or any kind of antimicrobial agents to kill microorganisms within the biofilm, right? Then quorum sensing inhibitors. We talked about quorum sensing, right? Can we use certain molecules which are going to stop the communication between microbes? So if they cannot communicate, therefore they cannot form biofilm, or they can continue to establish that biofilm-like structure where you can easily eliminate them. What about enzyme? Right? Can we use enzyme to break down the biofilm? Can we? Answer would be yes, right? Because the molecules we mentioned, polysaccharide, protein, lipid. If we have enzyme for those target molecules, you can easily disperse them, right? You can destroy biofilm. There are a lot of natural and synthetic antimicrobial peptides. We'll try to use some of those examples. Antimicrobial peptide or plant-derived polyphenol or essential oil, some of those are antimicrobial, right? They can also inactivate microorganisms. You heard about CRISPR-Cas, right? CRISPR-Cas9 is a gene editing tool, right? So CRISPR-Cas, it finds the gene target, and then enzyme Cas9 that can chop that, cut that section. And a lot of times that can be manipulated in a way you can control microbial growth or biofilm growth. I'm not going to spend any time talking about it. These are new technology people trying to use to control or prevent biofilm. Nanotechnology. Can we use nanotechnology approaches to control or prevent biofilm? I will show you some data where we used and other people also have used, can be applicable for controlling or preventing biofilm formation. Okay. Probiotics, again, we talked about it. Probiotics can compete with microorganisms. They can also help reduce biofilm formation. Even though they themselves can form biofilm, but they can still help remove or prevent biofilm formation. What about this one? Biophysical chemical therapies. There are some fascinating research in progress. If you think about wound, right, wound surface, and wound have typical biofilm, right? We discussed about that. And there are also nanotechnology based approach where you can on demand deliver antimicrobials. So that means your sensor can sense the environment, okay? And then strategically you can ask your sensor or device to deliver antimicrobial agent. One of the applications is the ozone. You heard about ozone, oxygen, O3, right? Ozone, and it's highly antimicrobial. And ozone, of course, we have the ozone layer on the top, right? We are depleting ozone layer, and that's the reason we are getting a lot of sun ray coming through. And that's part of the warming trend we are seeing. So we have to protect our ozone layer on the environment. But here in the ozone, we use ozone as antimicrobial or sanitizing agent. And so some of these wound applications, I don't have any data or paper to show you, but that's why I'm trying to indicate that if you have an ozone molecule and you can strategically deliver ozone onto the wound surface or on the biofilm surface, you can control that. And I thought these are some of this fascinating ongoing research we are doing, or some of the people in engineering, they are doing this. If you got general idea, because I'm not going to present any data on this, but just trying to tell you these are the new generation of research in, we call it smart sensor or smart technology. It's on demand, will deliver bioactive molecule so that you can control the microbial growth, okay, on surface. And these are specifically designed for a lot of this biofilm in the wound surface, on the skin damage, okay, external. So we're going to talk about individual items briefly. So disinfectants, right? I mentioned that chemical agents, enzymes, probiotics, bacteriocins, bacteriophages. Bacteriophages, I didn't mention that in the graphics. Nanoparticles, QS inhibitors, and also called micro nano bubble. Everybody heard about micro bubble? Heard about nano bubble? Anybody? You did. The micro bubble or nano bubble, I'll show you the schematic. You know, it's literally the bubble. The water bubble forms, right? And then when the bubble bursts, it has a certain energy that can kill microorganisms. It's fascinating, right? And if you have a nanostructure of that bubble, it can penetrate in certain places in the microbial community, and then it can also kill them. So it's a new generation of processing technologies people are using it. So I will give you an example. Ultrasound, photoradiation, okay? All of those are done. So food industry, so disinfectants kill or inhibit microorganisms that can spoil food. Can be indirect or incidental food additives used on food contact surfaces. Applications are regulated by EPA, so you cannot use disinfectants randomly. Usually they are regulated. And some of them are used on raw meat or poultry, especially for washing, surface washing. We'll use a lot of like a low doses of chlorine, for example, right? Or ozone, I mentioned. And they can be used, or acid, okay, can be used to control microbial growth on surfaces, right? If they are on the meat surface, can you use this washing to remove the biofilm, okay? And so these has to be food grade, because we are all going to consume that afterwards, right? And of course, we wash them to remove residual disinfectants, but still, if there is any residual left, that could be a problem. And so these are the molecules, the treatments I mentioned: sulfide, sulfur dioxide, chlorine, ozone, quaternary ammonium compound, that's called quat, peroxides, like hydrogen peroxide, they use that. And these are the chemicals or disinfectants are used on surfaces to inactivate microorganisms. Factors that may affect surface disinfection. So we are trying to kill microorganisms, right? So if there is a biofilm form, of course, that may affect depending on the level of biofilm formation. What type of substrate you have, the type of microorganisms you have. or what are the conditions, external conditions. So all of these factors, I didn't mention that, you can look at it. If it is a biofilm, you want to know how old is this biofilm, right? If it is a newly formed biofilm, it's easy to remove, right? If it is a very old biofilm, I showed you some of the pictures, it's very difficult to remove them, right? So the age of biofilm is going to be problematic for how effectively your disinfectant can work. Everybody following me? So these are 4 factors. Within the factors, you can see, read it carefully, how much EPS molecules produced. Is the quorum sensing active? Persister cells. If you have a persister cells, they may not uptake any of this disinfectant or antimicrobial. I know you cannot read this. Can you read this from the back? Very rarely, right? Or unless you have a very good eyesight. I know I kind of feel bad that when the slides are smaller, I never like that, okay? But these are actually came from a paper, so I couldn't do anything. I didn't want to manipulate it. But just trying to give you an idea, what are those factors affect disinfectant? Disinfectant may not work. So pay attention to this. Degradation of degradative enzymes, neutralizing chemicals, microenvironments, horizontal gene transfer. So everything about, we talked about biofilm characteristics. All of those factors can affect the efficacy of disinfectants. Okay. Substrate, we're talking about substratum, means the surface, surface charge, surface topography, wettability, conditioning film, sediment accumulation. So where the biofilm is formed, right? You need to know the surface chemistry, surface properties. All of this is going to affect if the disinfectant is going to work. And this is extremely important, because if you are assigned to take care of safety of food or molecules or drugs, anything produced in that environment, if there is biofilm forming, they're going to ask you, how do you get rid of this biofilm? And then you are looking into all of these factors. As an expert, going to tell that, well, some of these conditions you are using. So whether you are using stainless steel table versus wooden table. Okay. So if you are preparing food, it's a practical application. I'm trying to use it. Which surface is most desirable for application of disinfectant? And what is that surface we normally use on the food industry or pharmaceutical industry or anywhere you do? The steel is the best preferable, right? You can easily sanitize them, and their surface generally discourage formation of biofilm, right? The way the structure is. But if you look at the wood or plant-based or any other surface, that may be favorable for microbial colonization, right, and biofilm formation. And if that happens, then you cannot wash them readily, right? And you cannot clean them properly, and then the wood would be reacting or responding to the chemicals or sanitizers you are using, right? So the surface topography, it has to be smooth, right? If it is rough, then chemical may not reach in every site, okay? I'll give you an example. And even if you have a scratch in the nanoscale level or microscale level, that still can prevent your disinfectant from getting into that area. So that is the herborage site where microbes stay and grow. And this is so fascinating. You don't feel it, but that's the true application. A lot of people spend their entire life trying to figure out all of the surface chemistry, surface topography, how that can help the biofilm to persist for extended period of time. So that become a continuous source of infection or infective agents if you are producing any food or if you are producing any pharmaceutical drugs or molecules, or a hospital setting, clinical setting, where is this pathogens coming from, okay? And what this is saying is that this is the only tool you have, actual powerful tool in your hand to get rid of a lot of those biofilm or biofilm cells. Everybody clear on this? Okay. So when I mentioned that the substrate is going to be important, Then also the bacteria, what type of microbes you have. What did we learn? Even if you look at Staph aureus, right? And if you take a multiple different strains of Staph aureus, and you already know that the biofilm characteristics may depend on the strains, right? Sometimes maybe serotype. Not, I also showed you data for Listeria, right? And Listeria, you can see some of them form strong biofilm, some of them form weak biofilm, right? So within the same species, you may find variation, right? And you need to know which organism you are trying to get rid of. Is it Pseudomonas versus E. coli versus Salmonella? All of those different organisms has different criteria for forming biofilm. Some form strong, some form weak. By now, you all know that, right? And you need to take into account when you are trying to apply this disinfectant. Then also surface charge of the molecule, right? Or the bacteria or the microbe. What is the surface charge of bacteria in general? Is it positively charged? Is it negatively charged? Charge-wise, what's your knowledge? Negatively charged, right? Okay, those who do not know, bacterial surfaces are negatively charged. Okay, so if they are negatively charged, wherever you have a positive charged surface, they are going to form a strong biofilm. But if it is a negatively charged surface, then you know it's going to repel, right? And so can you create environment or surfaces that you have that type of charge distribution? Now you can prevent biofilm formation, whether it's in the catheter or medical devices or on any surfaces. Okay, so that's why I'm saying today's lecture should be the most important one. You learn everything, how they form, what are the foundation, and now we're trying to get rid of them. We don't want them. What can we do to prevent it? Shape and size also important. Appendages. Appendages means what? What does our appendages mean? You all know that, right? Appendage, that terminology, English. Appendages means arms and legs, right? The bacteria has arms and legs, right? Come on, we talked about, right? Flagella, right? Fimbriae, pili, all of those are called appendages, right? So this terminology they use here, they're saying that appendages. So some are expressing fimbriae, they will be more attaching, right? If they're producing flagella, they will be going and attaching themselves, right? So all of those factors contribute to the type of biofilm we are going to see. Motility, microbial metabolites, tolerance and resistance mechanism to antimicrobials, right? All of those factors are going to contribute how strongly biofilms they're going to form and whether this disinfectant is going to work. The last part is the external conditions. We all know that, right? Temperature, pH, humidity, nutrient load, ionic strength, hydrodynamic conditions, antimicrobial agents. So these are some of the external factors, right? If the pH is low, a lot of times they may not colonize, right? If it's high, neutral, oh, well, perfect condition. They can grow, multiply. Same with the temperature, right? Today is cool, but 2 days ago it was pretty hot. So if you have a surface or surface exposure to microorganisms, so they would be growing much faster, right? When high temperature. And so they could form biofilm quickly and rapidly. So if you have a dense formation of biofilm, the disinfectant may not work well that day. Or also the function of disinfectants or chemical sanitizers, a lot of those are temperature dependent. Did you know that? So what temperature generally do we use to apply disinfectant? Just a common sense. So if I give you cold water, ice water, right? Or I give you warm water, which one is going to work best? Common sense. Warm water, right? Because that's going to dissolve all the chemicals properly, right? And it has a greater access to the microbes. Again, these are all common sense. So if you put sanitizers at low temperature, it's not going to dissolve, it's not going to diffuse well, it's not going to make a good contact, right? Because that's a low temperature. Your surface properties of the microbes also going to change. They're going to behave differently. Are you all with me so far? Okay. So all of those factors play a role how well your disinfectant or any antimicrobial is going to work. So I spent time on this kind of this setting up the foundation for you to understand. Okay, now let's talk about enzymes. I think all of you, if I ask this question, even if I don't go with this slide, you should be able to answer it. We already established what are the metrics for biofilm formation, right? I just mentioned that. You have polysaccharide, you have protein, you have lipid, you have DNA. So name one enzyme for each matrix. Polysaccharide, you got a lot of different enzymes, right? Carbohydrate breaking enzymes. Why not use those enzymes to break down the carbohydrate? Protein, protease, right? You got varieties of proteases. Use something broad spectrum protease rather than narrow spectrum protease, right? So you can break down the protein. What about lipid? Lipase, right? So why not use lipase to break down the matrix? Then DNA, easy, right? DNase, break down the DNA. So that's how they are trying to apply enzyme to break down the matrix. Therefore, the dispersion happens. Well, these enzymes may not be able to kill the microbe, right? They cannot. Generally, they don't, or they may not, right? Unless these enzymes are integral part of the structure. So obviously, you have to use something else. So a lot of times, we use enzymes together with disinfectants or antimicrobials, so that the drug penetration would be much easier and faster to each of those organisms. Again, all of these are common sense. I want you to use common sense. Don't think too critically. So maybe there is a more important in-depth or deeper questions to answer. But if you look at this, proteases, cellulases, polysaccharide depolymerases, alginate lyase, these are all of those conditions you can use to break it down. So if you look at these polysaccharide breaking enzymes, they have listed few of them. Okay, again, go over this slide. The DNA, then protein, I mentioned this. Then also they used acylase, we'll talk about that. That's going to break down the quorum sensing molecule. Again, that's the enzyme, right? Then lipase, and then also cellobiose dehydrogenase. So any other molecules you have, it can break it down. That works, right? Okay. Antimicrobials. So I may go over the bacteriocin a little bit. You already have general idea about bacteriocins. Many of you, right? You heard about it. So bacteriocins are small molecules or peptides. These are always protein or peptide, and they are going to be released by certain organisms. And usually the history behind bacteriocins is that when microbes growing in a community or in an environment, they want to compete, right? For the competition purposes, they will secrete some small molecules, and then they can kill their competitor, right? So if you have an environment, they are all growing in this community, and say, another definition you have to remember is that they are effective against closely related organisms. You heard about that? Closely related organism. What does it mean? If I'm a gram positive, I will work against other gram positive in that environment. Okay? And if it is closely related, then it may not work against gram negative. And so what they're trying to do is that in a given environment, all gram positive organism wants to grow. And so to outcompete them, some of them would produce this bacteriocin historically. So they get an advantage in that environment. They will grow, they will utilize nutrient, and they get an upper hand. So it's killing other gram positive organisms. Very rarely we will find broad spectrum except the reuterin. Somebody mentioned that yesterday. Other than that, actually they are very narrow spectrum. You can make some bacteriocin against other organism work is because their target is cell wall. Okay, I'll tell you a little bit later, or the cell membrane. And a lot of times in gram negative organism, bacteriocin may not reach to the side. So sometimes if you break down the outer layer, this is what we are discussing, chelating agent or hydrostatic pressure or injury, that time bacteriocin can get into the unrelated organism, can kill that cell. So you have to do some special treatment to be effective. What they do pore form, inhibit protein synthesis, also degrade RNA. Okay, that's the general function of the bacteriocin. And there are a lot of review articles, a lot of people worked on it. Early in my professional life as a graduate student, I worked on this molecule, PDOCine CH. So you can see class 1, class 2, and class 2 again subdivided, and then class 3. It's based on the molecular weight, how many different types of amino acids you have, whether it's a hydrophobic, sulfur-containing amino acids, disulfide bridge. This is one of the characteristics, and these are the different names and different organisms. Again, if you are interested, you can study a lot of those. A lot of work has been done, and this has been used in terms of research wise. A lot of data available, a lot of publications available, but niacin is the only one is approved by the FDA, Food and Drug Administration. And I don't know why other bacteriocins are not approved, but nonetheless, this is the only one been approved. And if you look at the structure, it got some modified amino acids. Definitely that contribute to the structure. Anyway, so how does it work? As I mentioned that many of them are actually pore former. And if you look at the class 1, if niacin, what they will do, they will bind, go through the peptidoglycan layer, and then interact with the cell membrane, and the way it interacts, and then ultimately form a pore. So the pore formation basically very similar to the class 1 and class 2. You can see that integrate and then they form the spore. Okay, and this is when the pore formation happens, and then the bacteria eventually lose their intercellular molecules, eventually lies, right, or die. But the gram negative, because of the outer membrane, you have a barrier, right? The bacteriocin cannot get into the cytoplasmic membrane. So if you destabilize outer membrane in the gram negative organism, and then the bacteriocin can penetrate and can cause damage, so that you can have some limited death or limited inactivation of microorganisms. Very clear? So is it going to work well in the biofilm setting? It's going to face the same challenge as any antimicrobials, right? Or antibiotics we talked about. The diffusion barrier is there in the biofilm, right? They can be absorbed by the matrix, so may not, and the cells are inactive or dormant, so they may not respond well. So it's the same challenge as bacteriocins as antibiotics. Everybody follow me? The same mechanism or mechanical barrier they are going to face. So it may not work. So again, then you have to break down the matrix with enzymes. That's one strategy. Or you can physically destroy them, right? Then you can drive antimicrobials. Okay. So this is antimicrobial peptide, again depending on how and what level of depth of study you want to do. This one actually published with a collaborator from Purdue. We worked on together, and this student was working on melittin. This is a small peptide, and you can see how they cause the lysis. So she modeled it and how this peptide formed this pore, and you can see that integrate into the lipid bilayer membrane, and then can sit inside, and then you can see how that formed that pore, and that's called water channel formation by this protein peptide. So depending on what type of different protein or peptide, melittin I think from bee honey I think. And then there is another one from the frog skin. That's also another antimicrobial peptide. So because not only bacteria, also mammalian system produce antimicrobials, right? We talked about cathelicidin or trypsin, right? Also defensin. A lot of those are antimicrobial peptide and they work same way. Okay? All right, so they are going to form pore and cause damage. Okay, now let's talk about a little bit about the nanotechnology based approach. Again, I think I'm sure you have learned or read papers, right? Or some of you may be working on nanotechnology based approach. So what our goal is, if you can use nanotechnology or nano carrier, nano device to deliver antimicrobial peptide, right? So for example, nano liposome. We talked about that a little bit. Liposome is a lipid bilayer structure. You can have a multi-layered liposome. You can have a different sizes liposome. What you can do, you can load up antimicrobials within that, and under certain conditions, they're going to deliver this molecule, right? So if you have a biofilm, the nanoparticles, because they can disperse through small spaces, right? That's the advantage. Imagine you have a biofilm and only the small molecules can diffuse through, penetrate through. So the nanostructure, we're looking at very tiny, right? 10 to the power 6 nanometers the size, right? And that's the molecule we're going to use to deliver small molecules. Nano emulsion is also another one. You can use a oil droplet, and then you can decorate these molecules, antimicrobial for example, outside to deliver it. Also, this is another one, again, it was worked on at Purdue University with one of my colleagues, and he's a carbohydrate expert, right? So he found some natural dendrimer-like structure. So what he did, he loaded niacin in the core, and as you deliver, whether it's for drug delivery or for antimicrobial purposes for food preservation, you can deliver this, and this model actually allows slow release of this bacteriocin, slow release of antimicrobial. So if you don't have this type of delivery system, what will happen? If you just suddenly load it up, like without delivery system, they're going to disappear pretty quickly, right? Because other substance like enzymes, they're going to break it down, or it's going to diffuse everything at one point. But for most applications, you want slow, continuous, controlled release, we call it. So slowly, you'll be diffusing over time so that that antimicrobial effect can last longer. Okay? And that's why the nano scale or nano delivery, nanotechnology approaches are extremely important, are efficient. So that's with the nano subject. But then bacteriophage also can be used, right? Bacteriophage attack the bacteria, and they can attack and they can kill. These are actually, again, a couple of colleagues from Purdue, they isolated these two specific virus. So I got the picture for them, from them for my book. So if you look at this book, I think I used part of these pictures. So typical bacteriophage, you have a head, DNA is encapsulated or packaged, the tail, right? This is the tail, that's the base, and this is how they inject DNA into the host, right? And then the binding is extremely important, right? The binding protein is critical because that's what dictates the specificity. And also the protein they use to lyse the cell, okay? And that's how you destroy it. So we are interested in lytic phase of the bacteriophage action. And these are all different types of bacteriophages, okay? They are not one you saw is a typical one, lambda phase, we call it T4 lambda phase. It's a typical one, but you can see all different shapes, sizes of bacteriophage. They attack bacteria. So people are studying quite a bit on bacteriophage, because you can find the greatest application of bacteriophage in number of applications, right? So you can do it for treatment. Instead of antibiotics, can you use bacteriophage to treat a disease in humans? Yes, no? It's kind of weird. There are a lot of work going on. So if you follow the history of bacteriophage as a therapy, Russia is the one, I think Eastern Bloc, they started many years ago. In the beginning of this century, they used, not this century, the last century, okay? They've been using this for a long time. There are a lot of old literature. Unfortunately, that literature in Russian language, and most of the people not familiar with the Russian language, but they started using bacteriophage for therapy. Whether it's a diarrhea or other cases, they started using it. So now we are coming back and we use it again for treatment of diseases. And just briefly, you know how they cause lysis, right? So once it's being attacked by the bacteriophage, it's going to release their DNA, right? And what you want actually DNA to integrate, eventually they will multiply, they will repackage and burst open, kill them, right? But you don't want the lysogenic cycle because that's not going to kill the bacteria, right? It's going to maintain it. So you need to know the life cycle, and you can control the lytic cycle versus lysogen. Okay, that is a way you can do it. But most of the time, you prefer this to kill the bacteriophage. Okay, what are the factors? How does it work all the time? Is it going to work well in the biofilm? And if so, which cells are going to be killed? Okay, think about all the biofilm layer, right? Multi-layer. So let's start this on the surface layer. So that's where they're more exposed to, right? And they're going to be affected or killed, right? What about the cells which are dormant sitting inside? Less likely, right? They are not functional, they may not take it, they may not divide. So therefore, bacteriophage is not going to divide, and then this cycle is not going to be completed, right? So it's going to be fruitless, or it's not going to be fruitful, right? So it will be useless, kind of. So it's going to have a limited application. But then again, you're trying to kill them when they are actively growing, right? Actively dividing, that's when it's going to work. So in the biofilm applications, you might find, well, limited. So now let's see. You know your certain surfaces, there is a biofilm formation, right? And I want to use bacteriophages. How can we use bacteriophages to completely eradicate biofilm? Okay, so we talked about the problems, right? Now I'm giving you the question. I'm going to, as a student in my lab, I'm going to give you this project. Find out a solution for inactivating biofilm or biofilm forming cells using bacteriophages. And what strategy would you use to ensure that all the bacteria in the biofilm, they are going to be killed, so that I can eliminate the biofilm problem from my industry or food or what, a catheter or medical device. So I already gave you the answer. You already know the answer. Okay, I don't know whether you can explain it, but you already know the answer, right? So you need to make them what? Active. So first step I will do, I will disperse the biofilm, mechanically or enzyme treatment, right? And then what I would do, I will try to make them grow, right? I don't want persistent cells, I don't want dormant cells. If I can change their nutritional condition, right, I want to make them grow, make them feel happy. And when they are happy, then heat it with bacteriophages. You see this? That's when you kill them. Not when they are depressed and sad. Don't try to kill them that time. When they are happy, means the bacteria growing and they are physiologically active, and then that's when you add the bacteriophage, you kill them. Okay, now you eliminated that. Then your problem is that I want to continue to use biofilm for preventing, continue to use bacteriophage for eliminating biofilm. Even for disease condition. So like I want to treat it. Is it going to be useful or effective after certain time, certain period of time? So first time you had bacteria, right? Or you made this happen, so the most of the cells are dead. So can you continue to apply biofilm so that you don't have that disease reoccurrence? He said no. That's a good thought. Anybody else? Just say yes or no. You know how the answer is. So you need the substrate, right? You need the host. So once you eliminate, that's the problem with the bacteriophage. Once your target is gone, the bacteriophage, is it going to be there around? That's the problem, one of those. That means we have to have some level of bacteria always maintained so the bacteriophage can replicate. But if the host is gone, usually they are going to disappear too. So you have to continue to apply, right? So that's one of the challenges with the bacteriophage. Everybody follow that? It's not like antibiotic, right? Antibiotic is going to maintain in the blood circulation for a while, and if there's a pathogens growing, they are going to be killed. So you maintain that dose for a certain period. But bacteriophage, once the host is gone, essentially they are no use. They may disappear as well. Until next time your host is back, host means in this case bacteria, then the bacteriophage is going to work, right? Okay, conceptually make sure you think about that. Okay, bacteriophage application. So I already mentioned therapy, control, right? Prevention, you can do it, but again, that's the limitation you're going to have. You have to have your host bacteria. and sanitation. So all of those applications you can apply phage. Desirable properties of bacteriophages for food safety application. Usually it has a broad host range. Strictly lytic, we are looking for lysis of the target. Propagated on a non-pathogenic host. They can propagate on non-pathogenic. So this is another challenge though. So if you are trying to control Salmonella, right? So you have to grow bacteriophage in Salmonella, right? So that means if you are trying to market that, commercialize it, so you are going to produce large amount of bacteriophage specific for Salmonella, you have to grow large volume of Salmonella. You see this? That's a nightmare. Because if there's accident happens, if you are a bacteriophage producing company, and you are going to kill a lot of people with Salmonella, because you have to grow a large number. So somehow, I remember one of my colleagues, he's working on, Bruce Applegate, working on bacteriophage. So always they're finding, can you use a non-pathogenic host, or engineered host, so that they can harvest a lot of bacteriophage. And that's a challenge. Like E. coli 15787, which is one of those deadly enteric pathogen, it causes severe foodborne outbreak. And so if we are going to make bacteriophage for E. coli 15787, you have to use E. coli 15787 to grow it. And when you grow large number, and it's the people who are working in that facility, their health could be in danger. Also, if there is a exposure. Or how do you purify the bacteriophage so that you don't have any bacteria in the bacteriophage preparation? Are you following my logic? So that means instead of treating, you may reinforce that infection. So that's always a challenge. Keep that in mind. Hopefully, they are going to solve all those issues, but these are actual practical problems. We know the complete genome sequence, non-transduction, non-viral transduction, absence of any genes encoding pathogenicity. You need to have that. Make sure it's not carrying any gene for pathogenicity. Oral feeding studies show no adverse effect. GRAS approval, generally regarded as safe, has been approved, some of them. Sufficiently stable under storage application conditions. So they need to be stable in order to use it. So this table, again, you may not be able to read it. You can, the slides you're going to get it. What is saying actually, what are those bacteriophages now approved or you can commercially buy? Okay, these are some of the company marketing it. These are the product. Again, you find it again, Listeria, E. coli, Salmonella. These are the different bacteriophages they are using. These are the target organism. Okay, and this is the website. If you are interested to find it, or you can Google it, you can find that. So these are some of those commercially available bacteriophages they are trying to market. Okay. Okay. So as I said before, a lot of times the bacteriophage may not work in the deeper into the cells, and I already mentioned you want to disperse them, right? You want to apply enzyme, and that enzyme can break down the matrix, then you can apply it. So here is the example. In this paper, you can read it. Enzyme can break down the matrix, and then it allows the bacteriophage to get in, and then eventually target the bacteriophage and kill it. Okay. The other bacteriocin also same story. I may not be able to reach that. So hopefully enzyme or some of this treatment allow the bacteriocin to reach to the individual cell. That leads us to corrupt sensing inhibitors. You mentioned that communication stop. Okay, so how do you do that? Review. So the Lux operon going to produce this corrupt sensing molecule. Acyl homoserine lactone is one of the major molecule, right? And then it binds back, and then it helps transcription of the gene, and that can lead to biofilm formation, sporulation, becoming pathogenic, they become motile, and also sometimes there will be bioluminescence, right? I think you heard that lecture on that day about Vibrio, and so they can be bioluminescent. So these are some of the functions. So this is the function of AHL. And we also learned that it's a cell density indicator, right? At low cell population, they are not going to make it. At high cell population, they are going to produce the quorum sensing molecule. So this is the way they produce. They bind to the receptor and activate. This is in gram positive. I used the gram negative as an example before, just to review it. And so now again, this is kind of like telling you the same story. So we are using all those different applications to control the bacteriocin. QS inhibitors, if you look at the list of chemicals, and I know many people working on it, especially the plant-based molecules are getting very popular because these are natural, these are sustainable. Can you use these molecules to stop their communication? They're going to inhibit those quorum sensing molecules. So these are some of those list of chemicals. Some of this coming from grapefruit juice, right? These are acylase that breaks down the AHL, and also a lot of essential oil, thymol, carbachol from grape or eugenol. These are all of those come from spices and herbs we eat. So those also can inhibit the quorum sensing. And these are well established nowadays, and people are trying to use that. Can we use that for biofilm control instead of trying to use antimicrobial agents? These are more natural. If it mixed with food, probably it's not going to be harmful because we eat that. These are grass listed, so that worked very well. So people are trying to find out all different types of natural, grass listed, plant derived molecules as anti-biofilm agents. Okay. These inhibitors again work same way. Somehow, if you can degrade this, right, this QS molecule getting in, and also you can prevent binding. Can you use antibody to prevent binding or neutralize the QS molecule, right? So that's been proposed. Also, some of the speakers mentioned that. And this is the AHL homocysteine lactone. You can clip this part, then this become a non-effective or non-active or unactive or inactive molecule, right? So if you cut it out. So this is how essentially works. And these are other things also. You can combine it with the linkers antibiotics, right? And we know quorum sensing molecule, they're going to bind to the receptor. This is a fascinating way you can control it. We know quorum sensing molecule bind to the receptor before it's taken into the cell. So can you tag antibiotics with quorum sensing molecule? Because they have to bind, and then the antibiotic can get in, and they can destroy the cell. So this is another way you can do it. Okay. I talked about the microbubble, nanobubble. This is kind of graphical presentation. So if you create ultrasonic wave, and the bubble, the water bubble, for example, right, or surfactants like a soapy water, right, it forms these tiny bubbles, and so this bubble can change the shape, and at some point, this smaller one is going to be bigger, right, because the small one coalesce, they call attached together, and then fuse together and become large. And so what happens actually when this bubble bursts is called cavitation, okay, and that's an energy differential, and that can actually disrupt the cellular architecture, including biofilm. And this is recent, relatively new technology. I got this in 2025, but I know some of our colleagues also working on this called micro nano bubble technology. Okay, and this bubble, when you can physically form them, and then there is a time energy differential, you can allow them to burst, and that burst creates certain cavitation. That's what we call it. So it's an energy differential, and that can hit microbes, tiny molecules, in a certain force. They rupture, they break open. Okay, and that's becoming important because it's like a non-chemical treatment, right? But you are just using some physical energy, right? In this case, ultrasound, and you can create this, and then you can get rid of microbial biofilm or microorganisms. And people are trying to use that also food processing conditions, some applications to kill the microorganisms. I already mentioned some of our work. I will just last two approach we are using to control biofilm. So already I talked to you about tunicamycin, right? How tunicamycin works, antibiotic, and also it's a good biofilm buster, right? So at different low doses you can control it. Again, just to review it, this is the lipoteichoic acid attached to the peptidoglycan, and this pathway is regulated by this small molecule called tunicamycin, right? And so you want to prevent that formation of teichoic acid or peptidoglycan. As a result, you can prevent biofilm formation. right? And I showed you data for the structural attributes I mentioned, and also structure in Listeria and structure in Staphylococcus. And the most importantly, the morphology changes, colony architecture changes, and also the biofilm, right? So at different concentration, you can see if you go higher concentration, the biofilm formation become more dispersed, because we know the molecules which are responsible for biofilm formation, you can break it down. And so that means when you are using this molecule, I think a lot of people are trying to use this tunicamycin as a therapy because of the resistance to penicillin, right? Which is very common. And so for that, they are working in the cell wall synthesis in a different way, and people are thinking, can we use tunicamycin as a treatment for some of the diseases, okay? Because it clearly shows not only just the biofilm formation, it also affects other properties. We already mentioned that. And so here, you can also prevent their attachment to KCO2 cells, right? At different concentration, higher the concentration, low attachment. And it could be function of you are killing the cells, and also at the same time, they're not producing this peptidoglycan and teichoic acid, okay? That's the way you can control it. So other one we did is the called smart antimicrobial nanoparticle. I already showed you the structure, right? So why do you call it smart? Smart in this sense, I don't know how you use this word here in India or some other countries. Here smart means we are talking about intelligent, okay? Is it an intelligent molecule? It can go and find the target and bind and destroy it, okay? It's almost like a laser guided missile, right? It go and find the target and just specifically kill that. So I will use this, hopefully I can explain it very quickly. So in this case, if you design a chitosan based nanoparticle, chitosan is actually a, the shrimp or crab, there's a large amount of carbohydrate, right? It's kind of, people are finding a lot of use of it. A lot of medical application people are finding. And the chitosan, the way it works is that at large molecular weight, it's not antimicrobial. But at low molecular weight or certain molecular weight, it become antimicrobial. That's the properties that are very fascinating. And so what you can do, or people are doing actually, you can create this chitosan nanoparticle. You know how to synthesize nanoparticle. And then you can tag it with the antimicrobial peptide. Nanoparticle, antimicrobial peptide. So now you can put something, a target finding molecule, right? So think about the last one which talked about quorum sensing molecule, right? You tag it with antibiotic. Can you use something like that that can find the receptor and bind there? So in this case, actually, we used Listeria adhesion protein. I mentioned that, and Listeria adhesion protein binds back to bacteria. Okay, when it's secreted, it binds back. So we said, can we use this LAP molecule on this nanoparticle with the antimicrobial peptide? Can it go and find Listeria and kill it? That's why we call it smart. Okay. So we published it. Actually, it works because that's what I told you before. LAP is synthesized and it comes and binds back. That's the beauty. If it binds back, then if I load up any nanoparticle with this LAP, right, with the antimicrobial. The other thing we also did epsilon polyl lysine. And if you heard about polyl lysine, that also antimicrobial. It tagged that as an antimicrobial agent. So first thing, I think this is a postdoc, he worked on it, and he created, in this case, we use gold nanoparticle. Let me tell you first about the gold, and then we'll talk about chitosan. And so he created gold nanoparticle, and this gold nanoparticle, you can see clearly binds to the, with or without bacteriocin, it can bind to the surface of the Listeria. You can see coated with that. And so what we wanted to prove is that if you have a bacteriocin loaded gold nanoparticle, and if you look at the death, killing, and essentially it's inactivating all the Listeria species. You can see the clear zone. But now we have this one nanoparticle with the LAP attached to it, just to see if it can show enhanced activity, because it's going to find the Listeria, can bind it and kill it. And so if you look at this, I don't know whether you can see all of you, you have a actually increased zone of inhibition, little bit at least. Ivanovia also has the same function. You see a little bit. So if you look at this statistically, there is a little bit difference or higher killing. So the hypothesis we came up with, that if you create smart, find a target, it binds and it kills more. So those are the creative way you can use during your research. So, well, I got a system which can kill it, but can I enhance its activity? Because you have to find out they bind to certain receptor on the surface. If they bind it, they can kill more. This is a common sense application. This one, nanoparticle, so we are looking at cytotoxicity. If this nanoparticle, if it is accidentally consumed by you, is it going to cause any damage? You can measure this cytotoxicity. I already told you about cell-based assays we did before, right? You can use that to verify are they cytotoxic. So these are also, if it can prevent adhesion of listeria by treating this with the nanoparticle. So can it inactivate biofilms on the plastic? We use that on the plastic biofilm. And then also, this is 24-hour old culture. This one is 48-hour old culture biofilm. And you can clearly see this is 6-log reduction. I mean, it's a huge reduction by this lab or the smart biofilm. You can see this clearly is killing it. This is the gold nanoparticle with the lab, and then the pediosine attached. So this has a good inhibition, but this one has showing the maximum inhibition. So this created the smart means finding the receptor, binding to it, and is inactivating them. And other application you can do right on the plate. You can do it on the conveyor belt where actual problem is in the industrial setting. You can use that to inactivate biofilm forming microbes. And these are good ways to monitor it. So if there is a dark color, that means the sample areas are positive. Because remember, the biofilm can form in the places where you cannot use the sanitizers to inactivate microbes. And that's the problem, because they are living in a lot of different places within the food production industry. Okay, and so like I was saying that they are growing in certain environment, and your disinfectant cannot reach there. And so if it is a conveyor belt, this is a small laboratory scale, but if you think about long conveyor belt, have you seen that? I mean, this conveyor belt is used for transportation of food, right, in the processing plant. If they produce something, they are going over the conveyor belt, they are going to be packaged, and then they deliver, right? This is the whole system. And so a lot of times, all of those places, the microorganisms can be throughout the conveyor belt. They can form biofilm, and they become a continuous source. So the factory, what they do, they use disinfectant or sanitizers to keep the biofilm. Okay, and so here we are trying to prove that this can be used as a spray to control it. Last part, I'm going to talk about the chitosan-based nanoparticle. And so the student he synthesized it, he conjugated with the polyl lysine, and then you tested if you have a chitosan nanoparticle, just the chitosan nanoparticle conjugated to polyl lysine, which is the antimicrobial. We wanted to see if they inactivate microbes, right? And so you are measuring the minimum inhibitory concentration. So CHNP, chitosan nanoparticle, if it has a 25 to 37 MIC value, and here you can see very low doses, you can achieve the same killing, right? So that means conjugated chitosan nanoparticle with antimicrobials is more effective than the chitosan nanoparticle alone. Okay, this is just a proof. You can verify that. These are non-toxic, so we always measure that. And we are looking at the biofilm adhesion in this case. And so you are using chitosan nanoparticle on the cell line and try to find out if they are going to be toxic to the cells. But the most importantly, can it prevent biofilm formation? Can it inactivate free-form biofilm? So here we use Pseudomonas, Listeria, Staph aureus, Pseudomonas, Salmonella, and E. coli. And you can see this chitosan nanoparticle can significantly inhibit. Except Staph aureus, you get lower inhibition. But in most cases, they can prevent biofilm formation. If we apply it on certain surfaces, they can prevent it, right? We want it. And this, if you look at this, we are looking at violet stained, right? If it is untreated, you expect them to form biofilm, right? But here, when they apply the chitosan nanoparticle, they can inhibit. In this case, could not. that correlate with this data, and here also. But as soon as you have a chitosan nanoparticle, all the blue color gone. That means the biofilms are being eradicated or eliminated. So that's confirming this data. What about preventing mixed culture biofilm? So prevention, or excuse me, this is also preventing mixed culture biofilm, and we saw that it's very effective. Again, it's proved by that whole process. Staph aureus listeria, Pseudomonas listeria, and we wanted to see if this is effective. So it's clear means they are killing this mixed culture biofilm. So can it inactivate preformed, already biofilm formed? Can they go through the biofilm structure, can inactivate them? And that's where we saw less effectiveness. So there are some decrease, but some cases there is no result or no activity. So that's the bad news, right? Mixed culture inhibition is low, but you still get some inhibition. So that's one way you can inactivate it. I think this is, I want to just quickly go over that. This is for the seed. This is alginate-based antimicrobial coating reduces pathogen alpha-alpha on the seeds. So this is the seed architecture where the bacteria would be, and they use a lot of this technology to eliminate listeria and salmonella from the seed surface. You saw that on the cantaloupe surface. One of the clever things they did actually called brushing and removing this biofilm. So physically you can break it down. So what they did, this is our colleagues, they did that. They had a brush set up on this biofilm, and then they had some antimicrobials. And so what you do actually, you rub that surface, and then you try this application, and then if it removes it. So they were effective in always the biofilm. If you have it, you have to physically break it down and then apply your antimicrobial, and that can effectively remove the biofilm. So their study showed that you can effectively remove this biofilm structure. So these are some of those fascinating technology or approaches that can help remove these biofilms on the produced surfaces. Okay. Clinical approach, quickly, I didn't realize that I have the clinical as well. You want to take a break now? I know you guys have time for break anyway. Okay, let's take a break, say half an hour or so. I think you're supposed to have your coffee break or tea break, right? Let's have that. Then we can come back and I'll finish it. The idea is that we can finish it before lunch. But today you're going to have a little late lunch because I got the other chapters to finish. Okay? Okay, let's do that. Take a break, and then I'll finish the last part. This is more clinical application. Okay? Before you go, do you have any questions to answer? Or you can answer after you come back. Start thinking about your question. Okay? Let's take a break. I know there was a question how chitosan nanoparticle works, right? Essentially, we are creating a small molecule. It can go close to the bacteria. The chitosan nanoparticle, it already has antimicrobial effect. But if you tag it with another antimicrobial peptide, then you can get the double effect. That's what I was trying to show in that one. Okay. All right. So let's focus on a little bit. If you can have your attention, some of you. Okay. Are you back? They're still working on something. Wound treatment, right? Now I'm getting a little bit, give you overview of the wound treatment, what people here are using. So weak organic acid can be used. So the bacterial biofilms in medical application, right, or clinical applications, and can we treat infection, biofilm infection. Organic acids are used, but it's a weaker organic acid. You don't have to pour hydrochloric acid, right, or phosphoric acid. You're going to burn. So usually it's a weaker acid, acetic or citric acid. These are permitted to use under certain applications. Ozone, I mentioned that ozone also can help. Photoradiation, I have a slide I think I will talk about. Bacteriophages are also used. Ultrasound therapy. Ultrasound therapy, I think they're finding number of ways to use it. Ultrasound, you know ultrasound, right? At certain wave they are sent. And sometimes it helps the absorption of drug or break down the biofilm. Okay? And so it disrupts the biofilm in a manner. Now if you add your antimicrobials or drugs, that can penetrate very well. And so one of the applications would be, I don't know whether you heard about it or not, the blood-brain barrier. It's very difficult to deliver drug to the brain. Okay? So now they are trying to apply a low doses of ultrasound in a way it opens up those barrier. Now if you deliver the drugs, it arrives to the site. So in case of biofilm, so can you apply those type of wave technology, and that can help deliver molecules. This also can stimulate immune cells also. Non-thermal plasma, you heard about plasma? Plasma is not that plasma we are talking about, serum from the blood. This plasma is called cold plasma. So if you run, say for example, if you have a gas, right, and you run electricity and it ionizes the gas, okay, so it's ionic particle, right, and certain gas, if you run electricity, so if you're in the air, right, you can create, you have nitrogen, oxygen, all of those carbon dioxide, all of those gaseous molecules, and what it can do, it can charge these molecules to become antimicrobial, and it's a highly charged ionic molecule, ionic species, and that is called plasma. And plasma look like it's like a glowing, I don't know, I wish I had some pictures to show you. It will glow, it will change the color, it will look like a bluish application. The plasma, they call it cold plasma because you don't raise the temperature, and it can be very effective tool for food preservation. And what they are now using also for therapy, for treatment of some of the diseases by creating plasma. And so if you look at this called non-thermal plasma, atmospheric cold plasma generates photons, electrons, neutrons, and when they are exposed, they can constant supply of energy, and that's what actually inactivate microorganisms. This is again fascinating technology. Nanomaterials I already mentioned, but then you can also apply near infrared or other magnetic field to activate those nanoparticles. Remember, they are magnetic particles, and if you put it under certain magnetic field, they would move around, they can behave differently, they can stimulate cells also, okay, or immune cells. So there are a lot of applications in this area. This is a picture showing all the matrix. You need to disrupt it, then you apply your therapy, physical disruption, your microbial to disrupt the biofilm. That's the message all the time I'm repeating, right? Disruption of the biofilm. So if you have the wound, and already I showed you some of those, if you have a surface wound and already formed preformed biofilm or biofilms are already formed on the wound, can you deactivate those biofilm structure so that you can easily kill those organisms? And so that's where I think use the example of ozone. But if you use, in this case, weak organic acid, and you are making actually hydrogen ion, right? This is the function, this is the mechanism how it activates the making these hydrogen ions, and which can inactivate microorganisms. Okay, this acetic acid forming acetate. Read this review article. It tells you how these organic acids are going to work on microorganisms, which is the potential, right, of the membrane. Eventually, that can disrupt the microorganism. How the acid works. Okay. Bacteriophage you already talked about. Photoactivation, and photoactivation is kind of called light therapy. You heard about light therapy? They will apply certain wavelength of light, okay, and that activates the say if you are all using certain organism, and it creates the reactive oxygen species, which is highly antimicrobial. So the light energy converting into reactive oxygen molecule, and reactive oxygen molecules are usually very toxic, okay, that can kill microorganisms. And so they prove that blue light in the range of 400 to 470 nanometer, ultraviolet, but not mutagenic. Ultraviolet has again different one, A, B, C, different range. Okay, I think A and B more toxic than the C. This is the wavelength range, even though there's ultraviolet. So those light can be used to create these reactive species, and that can inactivate a lot of microorganisms. It's called light therapy. Okay. Therapeutic intervention against GI biofilm. I already mentioned you can inhibit adhesion, you can inhibit biofilm formation, you can eradicate mature biofilm. Again, chemical, enzyme, antimicrobials, right? And I think in this case, I showed you this slide before. They are using fecal microbiota to transplant, and then replace the natural microflora, and then restore the community. That way also you can manipulate the bacterial biofilm in the GI tract. Also you saw this, right? They use the colonoscopy and jet washing. So this is before maybe really bloody, and you see a lot of biofilm like structure, and then after you do that, you lose a lot of those biofilm like structure. So it can help remove physically the biofilm from certain areas of the GI tract. This is actually a lot of bacteriocins and a lot of inhibitory molecules listed. I know you cannot read it, but once you have this slide, you can go closely. And these are the applications for a lot of different treatment. These are all treatment for biofilms, okay, and these are for therapeutic applications. So those are the things I had in mind, and I think the question you had also is that how the ozone is applied to the wound. What I was trying to tell you is that there is a sensor that allow the understand the environment, right? So if you have a lot of microorganisms forming biofilm, and now you want to apply ozone, and what it does actually, it senses the level of ozone you applied, and it's going to remove the organism or kill the organism, and then it's not going to continuously expose it. So what people do nowadays, a lot of times they would carry ozone generator, and so you can have a large quantities of ozone delivered. But ozone is also toxic to you, right? You don't want to breathe, and therefore they want to do a more nanoscale controlled delivery of ozone. So the smart sensor can sense the level of ozone, and if it is saturated, then we will stop delivering. And what we are trying to do, the project, research project, the engineer trying to do, I collaborate with them. What they are trying to do is they develop this sensor like a patch, and you put it on the wound. So they are doing it in the pig surface. We have published some papers on the pig skin, and then you have this patch, and you can monitor the impedance, electrical conductivity of the wound, and that also tells you the pH of the wound or the condition of the wound. Then when you realize that a lot of microbes are growing, it's going to change the acidity of that environment, right? And so that it changes the impedance, and that's when it triggers the ozone delivery system, which is connected to that side. Then it will deliver very low doses of ozone for a limited period of time, and then it inactivates those microorganisms. It's like you are doing not continuously when it is needed over a period of time, many days, and that way it can help repair a lot of those wound tissues. And wound, you know, there are a lot of different types of wound can happen. diabetic wound, right? People who suffer from diabetes, they have a foot ulcer, the skin, they are susceptible to a lot of infection. So wound is one of these major, problem people suffer from, and you need to have a smart way to control that. And again, all are biofilm causing that problem. Okay? Any questions on this subject or the control prevention? So my goal was to give you, I know again within this short period of time you cannot give you everything you need to know, but overall the implant in your head, what are the major strategies, right? Whether it's a physical strategy to break open, then you can deliver enzymes or antimicrobials, right? That's what we are trying to do, disinfectant. But then you have to separate if it is a abiotic surface, right, versus biotic surface, right, or catheter, or all of the surfaces, then you have to have a different treatment. You cannot use the same treatment you are using on the food contact surface or stainless steel. You cannot use it on the human tissue, right? So you should be able to differentiate those two, biotic or abiotic surface, before you apply. Good. Okay. Hopefully this chapter gave you a broad picture of these biofilms and how to control it. Is there other one ready? Detection. You had it? Yeah. And you already sent the PDF to the students? You'll be. Okay. So they're going to receive these, right? Okay. Let's do this. Yeah. Okay, so last part of our lecture going to be detection, right? You already had some background information or knowledge. Some of the speakers already mentioned how we detect the biofilm. So what I've done actually in this section, there are specific tool or method you can use to detect just the biofilm. Then I'm going to spend also a lot of time talking about not only just the biofilm, the individual cells, right? Sometimes biofilm is a solid structure, right? And some technology may not be able to detect that. Okay, I'm going to go over that. But some cases, you need to break it open, right? You break open, get to the individual cells, and then can we detect those individual cells. But they're coming from a biofilm, correct? And this is the approach we can do. So when you are learning about this detection technology or detection platforms, it could be very, it has a broader application. Let's put it this way. Okay, you can use it for different organisms, different cells. Whether it's a prokaryote, eukaryote, also within the prokaryote, whether it's a bacteria, virus, fungi, or their byproducts or toxins, you can detect all of those. So I'm going to give you a flavor of all those different types of technology. So I'll start with conventional, then at the end I will talk about some of these biosensors or engineering tools, what people are trying to do. There is a period of time in my professional life, I spent a little bit of time trying to develop detection platforms, okay, for pathogens. And so I'll go over some of those, okay. So this is, I think I'm trying to give you a little bit about what type of research we do in my lab. So I spend quite a bit of time diagnosis and detection of microorganisms. So whether it's coming from biofilm or it is coming from a disease conditions or whatever microbes may be. But the detection tools are very common, right? Same plating method, whether the bacteria coming from a clinical specimen or it's coming from food, right? Or from soil or environment. They're all going to be same procedure at the end, right? If you're trying to use a sequencing, right? Sequencing is sequencing tool, right? You can use that sequencing no matter where the microbe is coming from. Is it clear? And so the source can be varied or variable, but the tool is going to be same. So I'm going to spend time on those tools, okay? So we spend a lot of time. So nowadays, we are not spending much time on this development part, but mostly I already talked to you about the pathogenesis. We worked on also prevention, bioengineer probiotic. I already gave you a presentation yesterday. And then other area, again, I'm not going to talk about is on drug delivery. But then you might say, well, it is a wide range of research program. Actually, this drug delivery research came from this pathogenesis. And if I have another time we meet, we can talk about that, because a lot of drugs we take, they are not absorbed through our GI tract, or very small percentage. What we are trying to do is that, can we develop the GI barrier, gut barrier, right? So we manipulate gut barrier, so the medicine you take orally, it will be absorbed more. That's what we are doing. So you see this, our research is all related. It's not totally different. So I work with pharmacologists and drug delivery people, and maybe we'll create nano carrier system. So if you want to deliver insulin or antibiotic, they will be absorbed at higher rate. I don't know whether you knew or not, most of the drug we take orally, what percentage of the drug is absorbed by your system and what percent goes out? Any clue? We take a lot of medication, right? Orally. And any guess how much of the drug actually absorbed by our system and rest of them go out? Any percentage? Just guess. Give me a number. 30 to 40. Okay. Anybody else? It should be good. I mean, good question, right? Have you ever thought about it? Because you remember, our gut is complex, right? We learned that. I was showing the GI barrier, innate barrier, right? The mucus, the microbes, the acid, the bile, right? All kinds of stuff there. Then food we eat, beverage we drink, right? All kinds of barriers there. And there you are taking a small pill, antibiotic pill, or diabetic pill, or any kind of drug medicine we are taking, right? We call it medicine, and you are taking it. How much of it is actually called bioavailable? Means your system absorbing it, getting to the blood circulation. That's too high. Anybody else? So now I gave a boundary number, right? The number you said is too high. Anybody else? 10? 10? Too high. Still high. See what we are getting to? Okay, let's say 2 to 3% or 1 to 2%. So that means you are taking anti-cancer drug orally, which is $1,000 a pill. $1,000, one capsule daily cost $1,000. And how much of the drug you absorb? 1 to 2% of the drug. Rest of it, where does it go? Go into the soil and water and sewage, right? Or the drain we talked about. All those drugs our body is passing through urine or feces, right? Out. So that means we are not efficient in delivering our drug, and most of the time we like it orally, right? You don't like to be injected or poked. And that's the difference. You can inject any drug, those are injectable, and their efficiency is pretty high, right? It directly goes to the blood circulation. There's no problem. But most of the time we don't want to be poked. We don't want to be injected, right? And you need somebody to inject it, somebody, either doctor or pharmacist or quack doctor, whoever, they have to inject it, right? Which is not convenient. Or you have to inject yourself if you're taking insulin. A lot of times we take our own insulin because you have to take it daily or certain period of time. So that's a huge challenge. That's a huge problem. So the whole idea being is that, can you control the gut barrier, epithelial barrier? And I showed you some trick, right, yesterday. The dysfunction, if you do it controlled way, you can open up the gut barrier, epithelial cells, and we figure out how to do it. This protein can help us. So that's how we could deliver drugs efficiently. So if you're taking antibiotic, instead of 2%, I said I could do it 10%. And that's a huge profit to the industry. You see this? So you don't have to make that much of drug, or you don't have to charge that much of price. So now you learn something new, right? 2%, very small amount of drug being absorbed. So the bioavailability is very important. No matter we take a lot of good food, right? You're saying the polyphenol, right? A lot of this good stuff, we take it, but you have to ask this question, how much it is really absorbed by your body? How much is used up by microbes in our gut? How much they get lost? That's a huge challenge. Other challenge we have, the blood-brain barrier. Whether you are working on Alzheimer's disease or trying to treat any disease related to brain, that's even the toughest barrier. Drug doesn't go. Can you open up the barrier to drug, deliver more drugs? So anyway, that's interesting. I'm learning a lot of stuff in this area. That's not my expertise, but we are doing certain things to help. And microbes actually showed us the path. Did I say you that? Microbes know how to infect brain. Did I say that listeria goes to brain? Can you follow listeria, how to get and add drug to that? Years ago, we published a paper in Nature Nanotechnology, and we used listeria to treat cancer or deliver cancer drug, because listeria knows how to go through the body. They're intracellular, they grow. So we targeted certain proteins, it went to the solid tumor, and then a drug analog, we put it there, and it delivered that drug analog into the tumor. And if you can kill those tumor cells by delivering specific drug for the cancer, that's how you can do it. So the microbes know the trick. Follow the microbes. Vibrio, listeria, a lot of the Shigella, a lot of those microbes, they know how to go around the body and follow that. That's what we've been doing. So that's how the good use of those bad bugs. Okay? All right. Example is the Botox also. You heard about the botulinum toxin? I talked to you about botulinum toxin is deadly toxin. But if you take it low quantity, you can control like a uncontrolled tremor. Have you seen people sometimes they're just sick? Their muscle, they cannot control it. And you can use this drug, the Botox or the botulinum toxin at low doses, it can stop the nerve impulses. So where did you get this drug? From botulinum toxin. Now, that kills people, but you get a good use of that. So you have to know that a lot of our therapy coming from microbes, the deadly bad bacteria or pathogen. Okay, that's always, hopefully, if you are trying to study microbiology, that should be inspiring to you. Don't think bad thing all the time. Don't try to find bad thing in people. Always there's a good thing in certain people, right? It's the analogy for bacteria. So some bad bacteria, but I don't get mad with them because they are bad. But you try to study them and try to take that as knowledge and try to apply for good or betterment of the humankind or disease treatment or therapy. Okay? All right. So what I'm going to show you, I think go over some of those techniques used for biofilm or pathogen detection. Staining methods, already some of those things being talked. Conventional culturing, microscopy. You already did some AFM yesterday, or not AFM, AFM, right? Yes. So you got some general idea. I'll show you some pictures. CRISPR-Cas9, again, I will see that if I have a slide detail on that. And molecular methods, PCR, genome sequencing, right? And then the biosensors. That's where I will spend a little time. So if you look at the staining method, there are different types of staining methods you could do to identify the biofilm. Crystal violet, that's the common one, right? Then you got all those propydium iodide, lectin, acridine orange, DAPI. These are all kinds of different staining methods used to look or identify the biofilm. For example, you already saw this, crystal violet. If you have a biofilm, you can clearly see that biofilm produced by this organism. So these top three showing strong biofilm formation, whereas this bottom three not showing much biofilm. So you can differentiate biofilm intensity by staining with the crystal violet staining. Okay, you already seen this, right? This is again crystal violet staining. This is one of the common one, widely used, and it's very simple, easy to use. Okay, then fluorescence, right? Then if you stain with acridine orange or propydium iodide, you can also find this cluster of cells. Okay, and then I mentioned about the stainless steel. So if you look at a stainless steel surface, it looks smooth, right? You don't see anything. But look where the bacteria hide. So if you have a scratch, just a thin scratch, in naked eye you cannot see that. Like whenever you are using knife or hard material on the stainless steel, it can make small groove. So if you're trying to wash it, what will happen? You cannot wash it, right? This bacteria are going to hide in that groove. Even in just small scratch, they can hide, and that's where they will form biofilm. So there is a small, then how do you drive the liquid through even a nano or micro structure, micron size structure. And usually you have to understand fluid dynamics. And that's why you have to really be an engineer to drive all of these molecules to these smaller hidden surfaces. Then you can take care of this biofilm. This is on stainless steel. Okay, this is an example. So they will grow on those places. And then if you look at actually the large number of techniques they use it just for biofilm, that's called roll plate, sonication or vortexing, actinic orange staining, stick plating, or alginate swab. So it's pretty much like a if you have a device that's inside the catheter, right? The bacteria are growing or biofilm formed, then can you roll that and then stick it on the plate, agar plate, right? Because anytime you can transfer those sample to the agar plate, they will start to grow, right? That's the normal healthy environment. And as soon as they grow on the plate, then you know that they are biofilm. Because they are coming from an environment, already you knew that they are biofilm, right? You can differentiate it. They are not growing in the liquid, they are not in the test tube, but they are going on certain surfaces. Then you can open that surface and stick it on the plate. Very simple. That's called the roll tube, right? You know that there is a cell there. The growing means you can detect them. And then there is also, you can sonicate, sonication, vibration, that will break open. You can take the cells, put it on the plate and growth. So these are all growth based, right? You can see that. And then also plate based, I think there was a discussion on Congo red. Do you see that? Plate, Congo red, if it is black color, that means they are producing polysaccharide, slimy. If they are not, they are making red color, that means they are not producing biofilm. So it's an easy way to differentiate it. This is one of the cool plating method, you can use it. Then micro titer plate, already I showed you this blue color. Then of course PCR and multiplex PCR and sequencing you can use to differentiate or to detect biofilm. These are more techniques, right? Again, all of those are culture-based methods, and then the PCR-based method. These are also some of these other methods. Go through this, each of them. You'll find it in the literature. There are tons of different tools if you're interested in using a lot of those different detection methods. Okay, very simple, relatively inexpensive, and you can identify if they're forming biofilm or if your technique you use to kill them or inactivate them, you can also monitor that, right? That's why you need the tool to detect, correct? You need not only for diagnosis or detection, but if you are using sanitizers, if you are using antibiotics, antimicrobials, you want to verify if your application controlling method is working, you need this type of tool to detect it, right? Okay. If I'm doing so like you broke down the biofilm cells, what is the simple way you can do it? So let's do it like if there's a meat surface, you have a biofilm, right? And you can grind it up. And then you put it in a, we call it stomacher, kind of blender, mix it if you have a sample. Then you put some kind of color change, enrichment. So if you add it here, and if there is a growth, they will change color. And visibly you can see that biofilm, indeed there's a bacteria, that's indication of certain organism, it changes color, that tells me that it's positive, okay? Sometimes then you take that sample, put it on the plate, right? You have a selective media, Congo red media, all of these different agar plates, you get differential identification. You can find out, what type of colony they are growing, and each of them indicative of, say, for example, black colony on this plate looks like Salmonella. The red or non-colored colonies could be E. coli, could be other gram-negative organisms. So those are the differential media you could use to differentiate what type of organisms you have in that mixture. Okay. Then you can also test their metabolic activity. What type of sugars they like. They may have a different sugar reaction. So as they utilize sugar or substrate, they are going to break down that into substrate. The product, which could be acid, and acid can change color. So essentially, these are like beautiful small strip, and each of them are considered as a tube, and it has a different types of sugar. So for example, it has a rhamnose, or mannose, or sucrose, or glucose, or dextrose. All kinds of molecules are there. That certain bacteria has affinity for certain sugar, right? So one will use that, but the other won't. So if you see this changes color to yellow, that means they use that sugar. Okay, the one did not use it, remained pink or colorless. And so this type of metabolic fingerprinting can be used to identify organisms. Okay, this is the plate based. You can use that. You can also use antibody called serotyping. You can use PCR to detect them. Also, there are some automated called fingerprinting tool, genomic fingerprinting. You cut it into small pieces. You run a gel. Then certain pattern you see it, and that represents certain organism. Okay, it's called genomic fingerprinting. You can do those, and sometimes you can do the whole genome sequencing. Right, nowadays it's becoming so routine practice. You do the whole genome sequencing, and then also you can do shotgun sequencing. Right, you do small fragment, then assemble together, then find out what organism. These are all kinds of different types of strategy people are using for identification. Okay, so those are like a conventional method, take longer, but if you want to do very quickly, sometimes we can use PCR method or quantitative PCR. You can use ELISA. You can also lateral flow. Lateral flow device is becoming very popular. It's antigen antibody reaction, right? It's also called pregnancy test kit. This is exactly the same principle. You add antigen on the top, it migrates, it binds to antibody. Then if it is positive, it will show you green or blue or whatever color it is designed to show. And if it doesn't show that, then it's negative. So it's similar to pregnancy test kit. Also, we use magnetic bead, magnetic particle coated with antibody or a receptor type of molecule. It can collect bacteria on the magnetic particle. The magnetic particle, the properties are, they are not magnet. These are called they are not truly magnet. Oh, I'm forgetting the terminology. But it has iron oxide and it remains in suspension. But when they are in the presence of magnet, then it become magnetic. Okay. Otherwise, they remain suspension. I'm forgetting the terminology. I will come back. So when you add this magnetic particle, because there is a core and there is a iron oxide, so it's not a total magnet, but it has a magnetic behavior when you put it in front of magnet. So the whole idea is that magnet with antibody, it can bind to the bacteria, and then if it is a 1 ml, 5 ml, 10 ml liquid, and put a magnet underneath, then all the magnet will collect near the magnet, right? Then you can throw the liquid away. So you capture the bacteria using magnetic nanoparticle or magnetic particle. And then you have this concentrated bacteria, then you can test them number of different ways. Whether it's a PCR, flow cytometry, cytotoxicity assay, a number of ways. Okay. There are some automated systems. It can run the liquid continuously. There is a magnet and magnetic particles. They bring that in that site, and you can collect all the magnet bound bacteria or bacteria bound to magnet, and then you can collect it, concentrate in a small volume. Then you can detect it. Does it make sense? You heard about that, right? Magnetic nanoparticle or magnetic particle. This is powerful because we use that. So if you have a large number of bacteria or low number of cells in serum, and you can run the serum through the magnet, right? It will collect all those. What they do actually a lot of times, the cancer therapy, if you are trying to remove the cancer cells or certain population of cancer cells from the blood, they can run that blood through this magnetic particle. It will capture the cells, and then you put the blood back into that human. Okay, so these are some of the therapies used. They use this magnetic particle approach to collect or target organism. Got it? And these are all because they are coated with the antibody. That antibody targets your cell, whether it's bacteria or virus or even cancer cells. Okay, so that's very powerful tool. Other thing is that I think the sample preparation become very important because you got a food matrix or you got complex matrix. Then how do you separate bacteria from it? So years ago we developed this tool. You can put your food sample, and then once the pathogens grow or bacteria grow, it can be filtered, and then you can detect them very easily. And so if you have like a bacteria from a food sample growing in this device, and then take this liquid drop, you can directly add to the lateral flow, you get a signal. Okay, so the whole process is not that long, but the bacteria needs to grow. It will take overnight. and then you can test it. So like this in this liquid. So these are some of those ways you can detect it. These are pictures for data showing that quantitative PCR. Everyone understand quantitative PCR versus real PCR? Or do you know PCR? What is PCR? Right? Most of you know polymerase chain reaction. We are amplifying gene, right? A quantitative PCR is that you don't have to really run the gel. You can quantitatively detect the amount of amplified product you made by adding the dye which binds to the DNA. So you can spectrophotometrically quantify the amount of amplified product, and it makes the assay more easy or convenient. And you can actually quantify how many amplified product or how many amplified DNA you got. That should correlate with the number of bacteria you have already in that sample or target gene. Okay, and so the qPCR works very well. That's why you'll get this type of graph. Okay, now microscopy is pretty handy, right? You saw all of those pictures before, right? This is a bacterial cell forming biofilm. You can look under microscope. You can take picture, right? Fluorescence microscope. These cells are treated with the acridine orange or propidium iodide. They're going to intercalate into the DNA, right? And then you can take pictures, right? Fluorescence microscopy. Scanning electron microscopy, that's also common. You have seen this. Confocal microscopy. Have you seen the confocal any here? Confocal essentially is a fluorescence microscope, but it does layer by layer. So you get a high resolution data compared to if you look at the regular fluorescence microscopy. If someone used confocal for this slide, you will see much higher resolution. And it's like almost like a 3D, three-dimensional cell. If you see 3D pictures. So this is supposed to give you very high resolution analysis for detection of biofilm. Okay. Already you saw AFM, right? Did they explain you how AFM works? The principle of AFM. So usually you still use a laser. Okay, there is a probe. Okay, then probe is going to move. Then as you hit with the laser, and laser energy that's going to control the AFM movement, right? Then AFM is kind of like a surface topography. It's probing. It's running through it. If it is a bump and all of those things, it can create a plot or picture or three-dimensional picture. And so this is essentially what you saw yesterday. And this is what the laser shows, energy coming in. This is the probe with the tip. And this is the biofilm structure or any kind of structure. And this probe is going to go up and down. And this is how it creates this picture. So essentially like a microscope, okay? But it's not a microscope, right? And so it is creating based on the laser energy and then how it goes up and down. And that creates the surface topography. That's what is used to report the data. So if you have a biofilm, thick or thin or rough, all kinds of ways you can monitor, you can quantify. Okay? And that AFM tool is used. So these are more pictures of AFM images. And you can see very high resolution also in this case, they use metal surface, and that also enhance the signal response. This is a powerful tool again, okay, for studying any kind of soft tissue imaging, okay, or the biofilm also widely used. These are also more pictures of AFM. These are different organisms, and you can also take pictures. Anyway, last part I'm going to talk about biosensor. And you heard about the terminology bio and sensor, right? So it's a combination of biology and engineering, right? And so why do we use biosensor? What is the purpose? Because all those methods I mentioned before, these are conventional methods. Sometimes they are less sensitive. The sensor, we use it to amplify the signal. That's the reason, right? So you have a biological marker, biological system, which we want to combine it with the electronic, right? And so if the antigen antibody binds, right, a lot of times you need a large quantities of antigen or large quantities of antibody to see that antigen antibody reaction, like a lateral flow or agglutination test or precipitation test, lot of those. But if you want to use sensor where you can say, well, if there is a 10 antibody molecule binding to 10 cells or antigen, and normally by all these other methods you cannot detect it, but if I can apply the sensor which can amplify that signal, now I can visibly see that interaction. Are you getting it? So it's all improving the sensitivity. So the reason we use biosensor, if you want to detect say 1 cell or 10 cell or 100 cell, which cannot be detected by all those conventional methods I mentioned, except the culturing, right? You can culture a single cell, it will grow up to become a million cell or 10 million cells. Now you can detect it, that's easy. But this one we're talking about without that type of growth, can you detect at low level? And that's where the biosensor tools became applicable or useful. And so I'll talk about varieties of ways doing it. So if you go to, this is how, when we started working on it, this is the way I tried to picture myself, that that's biosensor, how it's going to work. And I think all those modern movies you watch, right, they use laser or that kind of, some kind of, like I send a signal and you get back, right? Or they send, okay, run light through it, right? Then suddenly they can tell you, oh, you got a cancer, or you got this disease or that disease, right? You want to detect it. So my way of thinking is that when you are scanning product for price, right, can you find out if there is any pathogens in that product? That's without opening the box, without opening the product. Can you find out if there is any pathogens or any microorganisms in that product? Imagine that. Or if I want to know this water bottle, if there is pathogens or bacteria in that water bottle, without opening it, without testing it, can I figure out if there is any bacteria in this water? Can you do it? I want you to think. So this is close to 150 ml. I don't know how much is this volume. So you have certain E. coli cells floating in there. I'm sure there may be, depending on how it is made, but there is a specification. It's supposed to be 0 bacterial cell in this water, right? But if there is, or there are, how do I know if there is any bacteria? Can you come up with some technology that can instantly tell me if there is any bacteria in this? I'm not asking for what bacteria it is, but I'm just asking, tell me, that's the first question I'm asking. There should not be any bacteria in this water I'm going to drink, because the filter sterilized it or did something. There should not be any bacteria in this water. But if there is any, or there are any, can we detect it? Yes or no? You don't want to be wrong, right? Go ahead. Huh? Well, yeah, you can spectrophotometer. That's a good point. Yeah, you are sending light, right? If there are some bacteria, can we do it? But the problem is their level is so low, regular spectrophotometer could not be able to give any signal. Okay, so that's why you have to be creative. The same principle. Okay, you hit it with the light, and if there is a photon coming out, if you can detect it. But you are on the right track. That type of technology can be used. Okay. So I will see at the end, see if you can figure out some of those. A lot of times I think I can tell you that a lot of times there are some laser, you can run through it. A laser hits the particle. You heard about flow cytometer? Flow cytometer, right? That's right. So that type of technology you can use it. But here you are hitting the laser, and the problem is that there are so few molecules in that water, the diffraction, the light coming back or going through, okay, forward scattering versus back scattering, a lot of times the molecule numbers are so small, it's very difficult to detect it. But there are a lot of technology they are trying to develop, hit it with the laser, and the scatter, the light diffracts from the surface because of the protein, carbohydrate, right? All the structures we talked about, they're going to diffract light. So it's a basic biology, but you are hitting with the light, the photon, which is coming back. Can you detect that? Okay. So a lot of times people are trying to develop technology for that, and usually it's a laser based. But the question I was asking is that can you detect it, anything without opening the package? And that would be the ultimate future to me. So if she's running, checking the price, can we tell that Well, it has some bacteria, right? It has a pathogen. Then, of course, we are not going to pay for it, right? Because it has a pathogen. We're going to pay for that package? No. If you find out there's a pathogen, why I buy it, right? I'm not there to grocery buy pathogenic bacteria to kill myself, right? Okay, we're not going to do that. That is just a part of the futuristic thinking. And I don't think they're ever going to be realized that this type of technology going to exist when you are scanning for price. Can you scan for pathogen? Are you getting my view, my way of seeing things in the future? So that was the futuristic, but this picture we took it many years ago. I haven't seen that future yet, and that's why I decided not to work on it. That's a bad example. I don't want to be. But anyway, we don't have that, but can we have equivalent technology that can do this? Okay, let's look at this, carefully monitor it. So here, this individual, he's a scientist at USDA, and what they are looking at, they are running this apple, okay? And there is a light there, camera sitting on the top. Are you following me? So what he's doing continuously, sending this image, scanning this apple as it moves through the conveyor belt. So here the question is that can you sort apple based on the shape, size, and any kind of bruise, like apple fall from the tree, right? When you go to grocery store, do you buy any damaged apple? No, right? So usually those are sorted out. Sometimes people do it manually, but big companies, corporations, they don't use any manual labor. What they will do, they will use technology. And they will use this kind of technology called hyperspectral imaging. You heard about hyperspectral imaging? Okay, if you are not in that area or the field, probably you haven't heard. Hyperspectral imaging is that you take images with a different wavelength, different types of camera, okay, and you collect series of images, right? And then at the end, you use the machine learning, or you heard about AI. Everybody knows about AI, right? So what it does actually, it takes images of a lot of those, the different surface of that apple, and collect all the images, and then it has a database with that images, and then it trains itself. That's why it makes it artificial intelligence or machine learning. Once it has this database images, and when it recognizes that that image represents some artifact or damage, then it will tell you that this apple has a damage or this type of damage. So they have developed this hyperspectral imaging technology to detect any kind of abnormalities on the apple. But how do you apply for pathogen? How do you apply for food safety? How do you know this apple, when you eat it, you are not going to get sick? Can we use a hyperspectral imaging technology to tell you that this apple is bad apple, don't eat it? Can you do that? So that's the technology they are trying to develop. I think they really gone on and patented this technology. What they do actually, any apple have this type of, say, fecal material, for example. This is actually conveyor system. So the apple rotates, and they take this image. So the entire apple from all directions, they take image, hyperspectral imaging. And once they take it, and it looks like this. So if you have certain, this is leafy green. So if you have some damage, the fecal material on this apple or tomato or bell pepper, and so your idea being is that if there is any kind of soil, right, or feces when it falls on the ground, and you know there are going to be microbes, right, any kind of dirt there, and so you want to exclude them. And so what they did actually, if you have a fecal material, so it's animal feces, and planted on this leafy green spinach, and you can see these areas where you have this, they inoculated intentionally, and you can find those out. That's the area they have some kind of fecal material. And fecal material means you likely have pathogens or microorganisms. Then you are going to exclude them. So they are doing some kind of screening to remove the food which likely to carry organisms. But you may not be able to say whether it is Listeria or Salmonella or E. coli or Staphylococcus or Streptococcus. You cannot say the specific organism. But we all know if it is mixed with feces, a lot of times you are going to get bad bacteria or microbes. So you want to avoid it. So this is the big picture they are using. So that helps you to save or prevent a lot of foodborne diseases. with a simple screening. So my vision was, when you scan it, can you have that type of laser scanning to tell you there may be some hidden uh, damage, or organism may be growing, or some of those fecal materials could be an indication that there are pathogens or microorganisms. That is the whole idea. Okay, so that's on the plant side. What about meat, chicken? So what they developed technology, either hyperspectral imaging technology, scanning all those chicken, which is running in a high speed in a conveyor belt. And this factory, actually factory, they process like thousands of birds a day, thousands. And so you have to scan very quickly. And so you have real-time information coming from this chicken, and what we're looking for, any kind of damage. So if you have a lesion of the disease, right, on the meat surface, so there's a bacteria growing, or there's a virus growing, or parasitic disease, you would be able to scan the surface, you'll figure out if there is any kind of disease. And they can exclude that. So as it's moving through the conveyor belt very rapidly, and it's getting real-time information in the computer, and then it can say, oh, it got some kind of defect, it's bad for you, and then they will send that to a different line. And then they will use it for different purpose, or use that not for human consumption, maybe they will treat it with something else to inactivate microbes. So this type of hyperspectral imaging, in reality, you would be able to use it for food safety. Okay, this type of meat, for example, this is a different wavelength. You get these images, and it's called hyperspectral. Okay, and then the computer collects the information, training, then and artificial intelligence used to give this information. So that's a powerful tool. These are also for biosensor, biofilm. They use the hyperspectral imaging. You can see this, they are getting this type of plot at the end, and they can predict that there are biofilm formed on surfaces. So these are for biofilm, they are using hyperspectral imaging. Got it? And that's one of the powerful technology. It's a non-contact, it's just the laser, right? Imaging, it can give you a lot of information. And I like this hyperspectral imaging technology. It's again spectral based, right? It's a light. Okay. What about the big field, the farm? Can we use some technology to tell that if there is a problem in the farm? So, for example, they grow lettuce or cabbage we eat, right? Or cauliflower or spinach. All of those crops, horticultural food, we grow, right? And we harvest, and then it goes to consumer, right? If you look at this. Can we find technology to tell us there is a problem pre-harvest? Before you go to the field, there is a problem in the field. Okay? So you heard about drone technology, right? Drones everywhere now. They put a camera, right? Can you use this drone, Internet of Things, they call it IoT, and digital agriculture, they will image the whole surface. Now they are looking for abnormality. What type of abnormality would be looking in a field? In this field, for example, this is a huge field, they are growing lettuce for salad, right? What type of abnormality you are going to look for in this field? I think I have more pictures of that. What about wildlife running through the field? The day before they are going to harvest, right? Tomorrow we are going to go harvest lettuce, right? And then I found out there may be some animals running through it, because I can clearly see they damaged the field. Are you going to harvest that? Most likely wildlife, the pig, wild pig or wild animal, any kind, they may be carrying a lot of microorganisms, likely pathogens. And if they are gone in that field, and if you don't have an idea, you go and harvest, right? Next day, and you sell it, and then next thing you know, you are getting E. coli 15787 outbreak. A lot of people dying out of it. So the bacteria, where they came from, this animal probably contaminated it. And we don't cook lettuce, do we? No, we eat raw, right? Green. So if that's not going to be processed, and likely the pathogen is already there, and you're going to get it. And that's the fact, we are getting a lot of outbreaks happening with this leafy green vegetable. Can we use drone technology to help you assess that? So what the assessment is that you can, a lot of those track, So think about the drone. You run it through and get the information. Likely there has some problem happened, or there was a flooding overnight. But you cannot see it because these fields are acres and acres, huge area. You cannot see it. You got to drive around to see the field. If you look at some of those fields in US, I mean, this is the monster field. It's like one person is farming a 10, 15 acres of land or even more, huge, and only they use technology to do that. That way you can monitor it, you can get the real-time information, then stop harvesting. That way you can control it. These are for beef production, beef animal, and what they use actually, this is real research. They're using again this drone technology to figure out if any of the animals are sick in the herd. And if they are going to harvest meat tomorrow, bring them to the slaughterhouse, do we want to kill those sick animals? Probably not, right? When it is sick means it's carrying some pathogens. So you want to exclude them. Or if you want to use a therapy or treatment, you don't know how you are going to scan hundreds of animals. So the drone technology can help you locate that animal, then veterinarian can go and bring that animal or treat that animal. Because these are used for meat purposes, food purposes, okay? And so this is also being used. This is what I was trying to tell you. Look at this lettuce field, how big it is. And so this individual, he can real-time monitor his computer and get a picture of this field and figure out that this part of the field, some wildlife went through, and that may have contaminated the area, or the overnight there was a flooding. You can get real-time picture. And once you get that, then you can make a decision whether to harvest or not, or sell it to the consumer, or maybe exclude that area, so therefore you can avoid the foodborne diseases. So this is how you have to think of the future, the drone technology to help you understand how to control pathogens in the field. Got it? And then again, these are all hyperspectral imaging technology. Okay, so let's go back to the lab now. Instead of in the field, so you bring a sample, prepare it, process it, and then you want to use biosensor and then detect it. I will show you some of those technologies, what has been used, people are using. So you, as I said before, you need to have some kind of receptors or biomarker, and you call transducer, if you are like engineering part of it, and you're going to transduce the signal, and you've got to have some kind of amplification, and then signal processing, which is the image processing, like processing, that can give you some printout, like some digital number, right, that correlates to the number of antigens in the sample. So bacteriophage, can you use bacteriophage to detect bacteria? We already said that bacteriophage can kill the bacteria, but in this case without killing, or you can kill them, but can we detect it? Okay, let's look at this one picture. Here is that microfluidic chip with the electrode in it. Try to simplify this, right? So you immobilized bacteriophage, and bacteriophage are highly specific for their host, right? And so if you add E. coli, they are going to bind to this E. coli only, because this is a E. coli bacteriophage. It's not going to bind to staph, or it's not going to bind to lactobacillus or other natural organisms. Capture them, you can on this sensor chip, and this is a clever technology. Okay, just watch it. There is bacteria author. He added this bacteriophage. The bacteriophage has certain properties. He engineered the bacteriophage to emit light. Okay, so it's bioluminescence. Okay, now let's grow the bacteria, right? In that meat, it's growing, and the next day, after say 12 or 16 hours, lab video. Let's look at it in the right side. If it is positive, then hopefully I'll be able to play this video. If not, I will be disappointed. Oh, here you go. Watch. This is time lapse. What is happening? So here the time lapse camera setup. Do you see bright field, right? The oxygen dependent aerobic reaction, bioluminescence, and the bacteriophage injected DNA, and then the cassette which causes bioluminescence, and then light up. So if you have a simple technology, right, bag, if the bacteria growing, you add this bacteriophage, it attacks the bacteria that emits light, and then you know this is positive, right? You don't need any other testing. The bacteriophage can make nice. Other thing you could do, we call it fiber optic. You heard about fiber? You seen Christmas light? This long, I think, I don't know, in the US, whenever in the Christmas time we walk in the stores, they will have a thin plastic, right? And if there's a light there, it's glowing. The other microphone? Yeah, just use that. Is it better? Okay, so I don't have to scream. I'm losing all my energy by speaking so loud. Try to. Okay. So in this case, we are using called microfluidic biochip, right? It's a small design. You have an electrode. You can have antibody. You can capture bacteria. You can also monitor the bacterial growth on the chip, and then also you can lyse them. You can do PCR, all kinds of stuff. And these are nanostructure or sometimes microstructures within the device. You can see this. And then we are able to detect pathogen or bacteria using this chip system. Then we already talked about the cell-based sensor, right? You already got that. And so what I wanted to show you is that you can engineer a cell with the specific receptors. Once pathogen bind to it, then you can detect it. So you are going to monitor color, fluorescence, luminescence, impedance. So again, we are trying to use a sensor, engineering tool to amplify the signal. But in this case, pathogen naturally interact or infect host cell or cell line. And that's what we are trying to monitor, right? So here we did a called 3D cell culturing, 3D sensor, you can detect them. This is example, different types of toxin, you can detect that. These are also we created called MESIA. This is another kind of cell-based sensor, and where you have a cell line, bacteria binds, and then you can detect them also. This was designed for Salmonella enteritidis and Typhimurium. This is another one. This is a B cell-based sensor for Vibrio cholera. So you have a receptor on the surface, which is the antibody. It binds to the bacteria or target, right? Once it binds, it activates signaling cascade. You heard about it. And in this case, it's supposed to give you light. So if there is a reaction, and if this specific complex is formed, this complex actually produces luminescence or fluorescence, and you can measure it. And these are, again, you are engineering the cell to respond to the microorganism or the toxin. This is also another one for botulinum toxin. So the last part I'm going to talk about is called scattering technology. This is a technology we developed in our lab called light scattering. We call it Bordeaux. The whole idea being is that if you take Petri dish, with the bacterial colony, right? You have seen that, always happens. Can you use laser to detect each of those colonies? That was the technology. Okay, so let's say if you shine a laser, even this laser, the laser pointer laser, if you have a plate today in your lab, I can show you how this works. Very simple, and you can detect or identify it. Anybody has a petri dish? You have colonies? Okay, let's take this after the class, or you can bring that plate. I will show you how it works. Okay, it's a magic, right? So let's see this. Okay, it's working now. Okay, let's switch it. Can you hear me? Can you hear me? Can you hear me now? It's better, right? Okay. Let me put it in my pocket. Can you hear me now? Okay, you can hear me, right? So, you know who discovered Petri dish? That guy. His name is Julius Petri, right? He was working in whose lab? The German guy, Robert Koch, right? You heard about Koch postulate? You should, right? If you're a microbiologist, you should know that, Koch postulate, right? So the Robert Koch, he's the microbiologist. His lab, this guy worked in that lab. And he was trying to figure out how to culture bacteria. And if you read I published a paper, actually there is a history of how to, grow bacteria on plate, right? Then we use laser to detect it. And if you want, I can send you that review article. So this guy actually came up with this Petri plate. That's why we call it Petri dish. And whole idea is that this is one of those discovery in microbiology. I don't think we can beat this. Without plate, we cannot isolate organism, right? But that's the beauty of it. If you have a mixture of organisms, you put them on the plate at certain dilutions, each going to produce individual colony. That's how we study, right? And it's one of the powerful tools for discovery in microbiology. So that was discovered in 1887. That's when he was working in his lab. Anyway, so if you have these colonies and you have no clue which organisms they are growing, but in this case it happened to be XLT4. XLT4, we know that it prefers salmonella. They grow salmonella on the XLT4. But there are a lot of other colonies also growing. So how do we know which one is what, right? So what we did actually, if you have this plate and run laser through it, I think this is close to 635 nanometer laser. You can buy red laser, blue laser, green, all of those different wavelength laser. This is a green laser, you can use it. And if you send the light through it, and what we found out actually when you discover it, it gives a called beautiful scatter signature. Okay, if you run through this is a colony scatter signature, and I showed you before. So this is a mechanical engineering student, and this is Priya, who was a microbiologist. They were working together. So when you run laser through this plate, and you can see this beautiful image. Okay, and that's when you realize that just using laser, you can identify bacteria just simply running through the colony. And we published a lot of work on this. Then we created this, we built this engineering device. Of course, with the help of engineers, they built this. And you have a plate, you have a laser, and then you have a CCD or CMOS camera. The type of camera we use, digital camera, you put it underneath, and if the laser hits, you will get this image. And so let me show you this video, how it works. So you have a plate with the colonies, right? And then put it under this laser. And so what it does actually, you put that in machine, and then you have a colony, right? And so this system is going to map the type of colony on the plate, where those colonies are. And then eventually, this is the map of the plate, okay? And it's going to show you where the colonies are. Can you see these colonies? It's a picture of the plate with the colony. Now you instruct the system to go and take the scatter image of this plate. And what it's going to do is that start here, then take an image, go there, go through this colony. And this is called traveling salesman algorithm. Traveling salesmen who are selling stuff, they want to go quickly to each places, right? And so that's what they do. So we said, take picture of this colony, the scatter image. So now it keep collecting these pictures. You see this one? Then you can use the AI or machine learning. Again, that image processing I mentioned, they are trained. Now they are looking for that signature. If they find that signature, they will tell it, oh, this is Listeria, or this is E. coli, or this is Salmonella. Everybody clear now? And this is what we did, and this is what we see this pattern. This is a clear example. If you have these multiple colonies growing, you have no clue what they are, and then Listeria would look like this. Salmonella would look like this. Cigatoxin producing E. coli pattern. You don't need even digital system. You can clearly say that these are the organisms you have. And you don't need anything else. Laser, Petri dish, and even the image. Okay? So if you find it after lunch, probably I can show you. If you have a plate and we can carry this laser pointer. I'll show you, truly you'll see. Anyway, so think about this plate. It has two different organisms. Can you tell? Probably not, right? They all look same, these colonies. We have two different types of Listeria, we mixed it. One is monocytogenes, one is in aqua. Let's run it. So this is in aqua, this colony. This is Listeria in aqua. This is monocytogenes. This is monocytogenes. This picture is not that clear, but it's based on the pattern. In aqua, this is monocytogenes. You can verify it. Pick the test, you can color differentiate it. So even if you have a mixed culture, you can detect them. What about sample from liver or gut, and you can run this and figure out if there is this target organism in your mixed plate. So it's a powerful tool. We had a multiple applications. This one I tried to show you earlier with the antibiotics. This is the normal pattern. Question I was asking, again, I think it's not working, right? That's okay, I'm almost done. The question I was asking, that this bottle, if it had a bacteria in it, right? And by now, I already had some drink, right? If it is a bad one, real bad one, I will not feel sick now, maybe several hours later, right? But what this technology was developed, there was one technology developed, they can detect bacteria on the in the water, but they will use the same laser, okay? You use a laser from different angle, and you collect the scatter signature, then you can detect up to 10 E. coli cells in the water. And I haven't seen the technology made a big splash, because a lot of times it's highly sensitive, and if you are a food company, you don't want to buy any of those sensors. Do you buy this? Morally, you were saying, I'm making large amount of food, right? Name a food product, whatever you buy in the store here. And I developed this technology, it's very sensitive. And you can find 5 bacterial cells or 10 cells. Then you're going to report that I have this bacteria in the food. And can you sell it? Probably not. So it's a loss, right? So why would you use this technology? Who is going to use it? Are you going to use it as a consumer? Probably not. Come on, you're going to pay that much of money to buy this device? $20,000, $25,000, $50,000 unit? No, you don't have that resource. So who's going to buy it? So we can do a lot of fancy research. Who's going to buy it actually, real life? So the medical field, there you might find some application. That's for the life and death. A lot of times we don't equate that in the money. So anyway, that's one of those problems we face with a lot of these biosensor technologies, right? So I think that may be the last slide. Let's see. Um, so you can apply a number of different applications: pathogen detection, indicator bacteria. You can study bacterial community, effect of sanitation, or like we talked about the disinfectants, how to control biofilm. If that has been eradicated, you can verify it. Molecular biology tools, you can, before you sequence it, you know what organism you have. A lot of applications actually. Anyway, so I just trying to give you a broad picture, broad view of these different types of biosensor or conventional techniques available. Hopefully it will inspire you a little bit, or if it doesn't, it's still, any information we get from anywhere, we store it in our brain, right? And you never know when you're going to use it. Even if you don't want to use it next day, tomorrow, next month, or next year, or some point, you might find it useful at some point, right? And so think about what I just talked. I'm not trying to solve the world's problem. And I'm trying to give you some ideas to inspire you to do certain things, right? So hopefully the knowledge you learn for this last 9 days, tomorrow is going to be 10th day, right? So I hope it was useful for you. You have any questions we want to answer before we go and take a break for lunch? I can go down there, so you can use my microphone to ask question. Anybody has questions? You can have any kind of stupid question too. So you can ask how reliable is it, right? We're saying that you have colony, you put a laser, and is it going to change over time? Is that a good question, right? Because you have a plate, right, colony, and colony is a dynamic system, right? Bacteria growing continuously. And why do we see different type of signature, the scatter signature? That would be a good question. Unless you are tired, you are not thinking clearly, right? This is a valid question. I didn't answer those things in my presentation. So the colony, when they grow in this little dome, and all those byproducts, whether it's a polysaccharide, protein, everything they are making, each of them eating different stuff, they're producing different byproduct, they're getting accumulated within that dome, right? And when laser hits, laser can discriminate each of those molecules present. So everyone has a different signature. So that's how you see a different scatter image. That's why you see these waves, circles, lines, all of this signature coming because of what are those organisms, what they eat, what they produce, and everything is in the dome. It's not diffused out. If it is growing in the solution, and when you hit it with the laser, it cannot monitor those byproducts. It can monitor only the cell morphology. Is it clear? And that's how we differentiate them. So is it going to evolve over time? So if you take it after 6 hours versus 12 hours versus 24 hours later, if the same plate, you're going to give me, depending on how old it is, you get one type of signature. And after some time, the signature may change. So usually you call it like a 4-hour window. That's when we try to detect them. And within 4 hours, because either the cells are dying or they're producing their autolytic enzymes that can degrade some of those protein molecules or carbohydrate, and that signature can drift or change. So you have to monitor over time. And we make a library based on that time window signature, very important. Also, we make that also based on the plate where they're growing. Because if you put them different food, different substrate, they will have a different signature also. So all those variables can be very important. Okay, any questions? No? Even a simple question? No? Yeah, he was asking that at certain specific dilutions. Yes, it's very important, right? If you have a crowded plate versus you have a fewer colonies, they're going to be a different pattern. Because if you have a crowded plate, means colonies growing close to each other, they can influence each other, right? Quorum sensing or any kind of other information. They can diffuse through the agar that can influence their growth pattern. So always we are looking at close to 100 to 150 colonies per plate. You can have a lower number than that, so you have to kind of dilute it in a manner you always achieve that number. That's very important. You don't want to have it too crowded. And also some bacteria grow fast. And you heard about motile bacteria? If the motile bacteria, how they grow on the plate? You have seen this? If you, those who are microbiology students, I'm not going to bother those who are peripherally microbiologist or not professional microbiologist, but amateur microbiologist. Okay. You know when they grow the motile bacteria on the plate, they have a motility too, right? So usually they're looking at a colony, you can differentiate the one which is motile, and the motile bacteria, we grow them in the soft agar, right? So that check their motility. Some bacteria, even if you don't grow in the soft agar, they can still move around, like what is that? Proteus, one of them, highly motile. So single to 1 or 2 colony can cover the whole plate. Those are bad bacteria for this technology. It's not going to work. Yes, motile bacteria, they would have a different type of colony, but you can manipulate the media in a way you don't want them to spread too much, so that you can get a laser signature. Any other question? So you have to have a proper dilution. No?
Sir, as we know, we use wax coating for increase of shelf life of food products. So what does it affect in our body when we consume that wax coating?
The coating wax. Oh, you're talking about wax. I don't have a whole lot of knowledge about wax, right? We know that wax you cannot digest it, right? Usually it's supposed to be neutral, and the reason we do the wax coating of a lot of food because it prevents spoilage, so the pathogens or other organisms don't grow, and it increases the shelf life of the product, right? And it stays longer. Generally, if you wash it in the warm water, it should go away, right? It dissolves the wax with the temperature. All those apple we buy, they are all waxed, you know. The cucumber we buy, a lot of produce we buy in US, these are all waxed. So they've been using that for a long time, and wax is considered it's the bee making the wax, right? And it's health-wise, it's supposed to be safe or neutral. It doesn't have a negative effect. And I don't know if it is a long-term effect, then they will not use it. So they've been using that for a long period, knowing that it doesn't have a toxic effect on the body. So I'm assuming it's neutral, will not have any kind of impact. Okay. Any other questions? Any kind of questions? Go ahead.
So what's the role of plasma in food preservation? Yes.
Remember the last hour we were talking about control and prevention, and one of the technology I was mentioning the plasma, and plasma technology is becoming extremely popular nowadays. This is actually what they do called generate the plasma. They run, for example, if you have a 2 electrode and you run electricity through this, and it creates spark, right? Have you seen that? If there is a distance, and that during that spark, what it does actually, if you have oxygen in the air, right? Carbon dioxide, nitrogen. So that creates this called reactive oxygen species. So the oxygen changed to O form, right? -1, so that's the reactive form. And once you have this ionic charged gaseous molecule, and that's deadly for microbes. So the ionic organ species can kill bacteria efficiently. And so the plasma they create by creating this electric charge, and that lasts for a while, and that neutralize or inactivate any microbes. And so this called cold plasma, cold temperature, it doesn't raise temperature, and they are using for a lot of food processing applications. And same time, if you have a wound on the skin and you want to eradicate biofilm, they are using that plasma also, that kind of a glowing color stuff, and they create right on the wound surface, and that way it can kill the bacteria. That's a good point too, but they have to have a dose dependent way, so you have a less called collateral damage, right? And so they try to control the concentration in a way it has less damage to your own cells. Yes, that's a good point. It does have, that's why you call it collateral damage. You heard about collateral damage? It's killing the bacteria at the same time also killing your own cells. Okay, yeah, that you have to kind of like a controlled way. You don't want to do too much, enough to kill the bacteria and stimulate the cells. Then there will be some damage. Yes, you have to accept it. but treatment-wise, it's much better than antibiotics, right? Or antibiotics may not be working. So a lot of applications they're finding it works very well, especially the medical devices. If you do that, there is no tissues, right? So you do that treatment before and then apply it. So catheter, a lot of these applications you'll find it also medical application is useful. But in the food application, of course, there is no worry, right? You have a food, whatever package you have, you created the plasma, it inactivate microorganisms in that food. Then after you open it, it's gone, but food is sterile. I mean, I won't say sterile, but it reduces the microbial load. Good? Anything else? Oh yeah, go ahead.
So you mentioned we can use bacteria in drug delivery. Drug delivery. Yeah, but the bacteria can also cause disease themselves, right? Then so we were using the modified organism or the wild strain.
Yeah, usually if you are using bacteria to deliver those type of biomolecules or drugs for treatment, that organism has to be modified. Yes, you don't want that organism to make you sick. That has to be engineered in a way you can knock off certain gene which is responsible for moving, or it can survive and deliver, and that's the end of that life. So you can create a lot of mutant or organism, engineer it, then you can do that. That's for live organism. But what if you don't need the organism? You can take the protein. For example, cholera toxin. You heard about cholera? It's a deadly disease, and also Asian countries we suffer from a lot of diarrheal diseases. So the cholera caused by Vibrio cholera, right? That's the organism. And it produces this toxin, cholera toxin. Also it produces 2 or 3 other toxins, okay? And so now what we are using cholera toxin for? The function of cholera toxin is open up the tight junction barrier. That's the junction, right? So now if we can use the cholera toxin at low doses with the medicine you want to deliver, including antibiotics, then it can open the tight junction very controlled manner, then help deliver the drug, right? In Listeria, all the data I was showing, the lab induces that pathway, so we actually patented lab. We already got the patent, and we talked to a lot of pharmaceutical company. So instead of using the Listeria or cholera toxin or some other toxin, can you use lab to deliver drug in your body? Because you know it opens. And then it closes after 4 hours. It has a reversible change. So once it delivers the drug, it closes and it works very efficiently. So we found out it can increase up to, I think, 15 or 18% more. Like instead of 2% I was mentioning. So we increased that. So we patented the technology. Right now the study is reviewed by Nature. We have submitted that, including structure and the application. So hopefully we'll have a favorable response very soon. And we're still looking for, we talked to a big pharmaceutical company. Hopefully they will come back and use it at some point in future. So everything we did, we are following Listeria. Like I said, I love Listeria for many years. We've been working on it. And now at get to a point, we're trying to figure out if Listeria can help us solve a lot of problems. And you know it goes to brain, right? Can we use that for drug delivery in the brain? So that's our next research. We are doing some collaborative work with Stanford and Purdue, and we have engineers and high-level young scientists trying to collaborate, pick their brain to help deliver drugs. So we are looking forward. But all of this came because we studied Listeria real detail. We tried to figure out what this protein does, how does it do, and all kinds of stuff. And so it's helping us in a number of ways. So don't think that these bad bugs are really bad. They are bad when they're into you, cause disease, but always try to think in an opposite direction. How can you take that bad properties and use it in your favor? Like I was saying, the botulinum toxin. Botulinum toxin is a lifesaver for a lot of different diseases. You know, I forgot the name of the disease. One of my colleague at Purdue, and he said, "Oh, my son suffered from this dystonia, muscle contraction, and only drug he has actually the Botox." I'm not thinking about Botox for cosmetic use. People use that, right, to look pretty. They don't smile. If they smile, their muscles are not moving. those who are taking Botox therapy, right? But it make you look younger, right? Your muscle tone changes. Usually the female, they use that because they care about the beauty more than the male probably, right? This is a gender joke, but they do that. But some male also, they do that. But I think I'm thinking from the treatment of diseases, right? Can you use a control level? And so there's a huge market for that, botulinum toxin. Hard to believe that, right? But in order to grow botulinum, you got to have a level 3 facility. Even I cannot grow it in my lab. Okay, so there is a special lab. That's where you have to grow, you have to use, you have to have a vaccination if you're working with this bacteria, Clostridium botulinum. And so that's the deadly organism. So anyway, the message is that you have to understand the mechanistic aspect and then take the properties from that and follow the bacteria, what they are doing, how they make us sick. Can you just use their, against their, to control them, and also use it for other applications. Do you see this picture now? Because the lab story I never told you, that opens, but we have a purified lab now, and a couple of students working on it. And hopefully, it will help us to deliver biomedicine, right? Or even pharmaceuticals, nutraceuticals, think about it. That can also open up. So a lot of applications. Any other questions?
So how does the presence of antibiotics and quorum sensing inhibitors affect the signaling and accuracy of biosensors targeting biofilms?
So you are mixing a lot of stuff together. Okay, first thing is that the quorum sensing molecule, they are secreted. So once they are secreted, right, and they're going to bind back to the bacterial surface, right? So the one I was showing you, that when you add antibiotics, tag it with the quorum sensing molecule, now it can bind to the receptor, and then the antibiotic can kill the bacteria, right? So now if you want to use it for biosensor, they're binding it, right? So can you immobilize this quorum sensing molecule on a biosensor platform? Whether it's a fiber optic I showed you, or microfluidic chip, right? Now the bacteria are going to bind to the receptor, right, in the opposite direction. Once they bind it, can you detect them? So the application-wise, you could use it, theoretically, you can use it for biosensor application as a detection tool. Like you use antibody to detect it, right? So antibody is your capture molecule. It binds, then you can detect them. Is that answer your question? The accuracy all depends on, no, that's a good point though. If the accuracy would be less, I mean, I would say accuracy means is detecting the bacteria, but thing is same quorum sensing molecule produced by one organism can also detect by other organism, right? So that specificity may not be there. That's the question I think you are asking. Yeah, but for screening purposes, you can say initial step, I could use it to see that, okay, this type of organisms might be present. But the specificity, yes, that's what you are converting into accuracy, but I would say more specificity. But accuracy wise, I would be thinking, oh yes, it is detecting bacteria, but is it pathogen? That's the accuracy, that's the specificity. So probably not, I think, because it's a broad range. Okay, any other questions?
What's the toxicological effect of nanocarrier molecule in drug delivery in reference to GI tract? As you mentioned, we only consume 2 to 3% of drug. Then we face so many side effects of the drug. Then when we target direct GI tract with the help of nanocarriers, then what kind of toxicological effect we can face?
Okay, let's see if I can answer your question. So the question is, what is the side effect of those nanocarrier in the drug delivery, right? So when they design it, of course, they are always looking for nanocarrier which is biocompatible. I mean, I'm not going to use a silver nanoparticle. No, silver nanoparticle is toxic, right? Gold nanoparticle usually not toxic, but always the toxicity has to be tested before you use that as a drug delivery agent. Because otherwise, if it is toxic to start with, definitely you're going to see some toxic effect. So in this case, we are going to use Now I have to think about what is the biomaterial they're trying to use. Usually supposed to be non-toxic, biological-based nanomaterial. I know a lot of people are using plant-based nanomaterial, right? And a lot of those are supposed to be non-toxic, right? Because it's a plant-based. And that's why there is a lot of interest on plant-based nanomaterial. Chitosan is also, non-toxic because it's coming from shrimp and crab, and it's a byproduct of that industry. And usually that's non-toxic. And that become also very attractive drug delivery nanoparticle. So you do the testing beforehand. And other thing is also we're trying to do is that even though you are creating a non-toxic nanomaterial, but you don't want that nanomaterial to accumulate in your system also. Because over time we don't know what type of effect it's going to have on the cellular process inside the cell. That could have also, but you have to test those beforehand. And so in our case, what we found out, even whatever research we did so far, with the nanoparticle, we want to test lab. Principle, lab actually doesn't go inside the cell, stays outside. It initiates signaling cascade. And so what we are thinking, nanomaterial tagged with the lab, and then it open up right on the surface. It just, with the payload, the load, it opens up the drug. It's going to pass through the barrier. Supposed to stay outside in the GI tract. And so that way, it should literally pass through the body instead of being absorbed. But you cannot guarantee that the cells are not going to take up some of those nanoparticles. In that case, if it happens, definitely we have to monitor, how we can control it, how we can track them, where they go, how much they accumulate. We have a system to track them, though. There are some technology available. It's in my head right now, but there are systems you can monitor where they accumulate. And these are some of those things that needs to be studied in detail. But nano carrier-based drug delivery in progress. A lot of people are working on it. And especially if you do a smart nano delivery, like the one we are saying, the receptor specific, then you can target with this cancer cell, like a colorectal cancer or other cancer specific, disease specific, cell specific, you can deliver that. And so we are kind of excited because it binds to certain receptor, because those receptors are expressed at high level in those diseases. Can we have an anti-cancer or anti-therapeutic drug or therapeutic drug attached to that? They will deliver directly into the cell. So those are the target-specific delivery system. But all of those factors you said, those are true. You need to monitor them, toxicity. That's going to be the future. Okay?
Sir, can you use the CRISPR-edited bacteria for gene transfer?
Yeah.
Yes, sir.
The answer is yes.
Yes, sir. Like the listeria you mentioned for to use the brain. Yes, sir.
Yes,
Yes, sir. There is some work going on CRISPR-edited vaccine production also for some diseases.
Listeria, we haven't done it, or I don't know anybody's doing it. But listeria, we use it for drug delivery or anti-cancer drug delivery into the cell.
Yes, sir.
They have engineered them. The company can sell it. I don't know. They use the CRISPR-Cas. Now CRISPR-Cas can be used to.
Yes, sir. We can knock out the virulent gene and use it. Yes, sir.
So for example, we want the bacteria not move from cell to cell, but take away its toxin. Okay?
Yes, sir.
So that it doesn't damage the cell,
Yes, sir.
but it moves. So you can customize.
Customize
Know
it.
a lot about it.
Yes, sir.
And so one thing I think they're doing, like Lactococcus lactis as a good vaccine candidate, because they're delivering it's non-pathogenic, and they put certain antigen for vaccine, and then it goes directly into the cell, and then it lies, and then delivers the payload or cargo into the cell. And that's what the cell use it for, whether it's a vaccine or therapeutic purposes. A lot of fascinating stuff going on. Real fascinating. It's so exciting. I think you got a bright future. You guys have to follow through. We pave path, but now you guys can take it to the next level. I think nano level, ultimate kind of like science fictional type discovery you can make, and so much to do, so much to work on. All of those problems, you just think about it and don't get distracted. Always focus on one thing. I think in my life, in my science, and I started with listeria somehow. I like the disease process. Always got back to it, and always trying to figure out how I want to learn more about this, go deeper. And I'm finding out a lot of applications. So the probiotic we created, that's patented. We are looking for, application. I showed you the story about that lady, she wanted. Again, all from listeria research, okay? Same way the lab we created, we patented it, and it patent went through very quickly. And so we are, trying to do towards that. Hopefully someday it will be useful for application for drug delivery, pharmaceutical industry. Like I said, I'm not a pharmaceutical, I don't have expertise on that, but we collaborate with professors who has tremendous knowledge in that. But I know about listeria, I know about this protein. What do they do in the biological systems? So for you guys also, think that way. Something passionate about you, it motivates you, stay with it no matter what, and then you'll find out that ultimate application, And you cannot solve all the problems. Just focus on one problem, okay? See if I can help improve that. That's how I would approach it. And think that way. Because you can get lost, whether you just recent PhD or you are in the PhD program, you are going to graduate, you are going to be faculty someplace or get involved in the research. Always you have to think that what is going to be my future. If you have an independent way of looking at it, so maybe this is the specific problem we want to solve. Sometimes it starts with your family. So if you say, oh, my mother had this problem and I want to solve this problem, that become motivation for them. Or my dad had this problem, or my relative passed away because of this cancer or this disease. A lot of times that become motivational force. And I know a lot of students start that way. And if you stay with that, that's your passion, that's what you to solve. A lot of people suffer from those type of diseases or conditions. You are trying to help them as well, And this is how I would start, and that's what you do, okay? And so that would be my advice for you guys going forward, okay? And stay motivated. That's so important. You have to love it. You have to like it. If you are not motivated, if you are not passionate, you cannot make a progress. It's always. And that's the criteria. That's the basis of being successful, okay? Any other questions? I know we are going 1:30 and hopefully that lunch is going to be ready, right? He was given the instruction to come late a little bit. Anything else? If not, let's take a break for lunch, right? And I'll be around also for a while in the afternoon if you want to have more questions or interaction or discussion. Any questions you want to ask is open. I'm open to you. I'll be happy to provide my input. I may know the answer, I may not, but I'll be happy. And also, I know you guys wanted to take pictures. Let's Doctor come back or Dr. Goyal or whenever or after lunch, right? That'll work, right? Let's do that. Okay.